Hello there, and welcome to Life Beyond Seen, where curiosity settles in comfortably and science moves at an unhurried pace toward rest. Tonight's journey takes us far back, long before cities, forests, or even dinosaurs, into a time when nearly all complex life existed beneath water. The focus is a question that sounds dramatic, but turns out to be surprisingly practical once you look closely. Why the Paleozoic ocean produced Earth's first creatures that could reasonably be described as monsters. Not monsters in the cinematic sense, not fire-breathing or mythical, but animals that were unusually large, heavily armoured, well-equipped for hunting, or simply unlike anything alive today. If you happen to be listening late in the evening, or perhaps early morning somewhere else on the planet, you might notice how strange it feels to imagine a world so different from the one outside your window. Most of us rarely think about oceans older than fish, older than trees, older than bones even. Yet those ancient seas shape the direction of life in ways that still affect every ecosystem now. Before we settle fully into that distant past, a small personal pause fits nicely here. Where are you listening from tonight, and what kind of environment surrounds you right now? A city apartment, a quiet suburb, countryside, maybe a long train ride or a late flight. It's an interesting contrast because the creatures we're talking about lived in a world with none of those human structures, just water, minerals, sunlight filtering down, and the slow processes of biology figuring things out step by step. And if you find this kind of calm, scientific wandering helpful for winding down, feel free to give the video a like or subscribe. It helps keep this quiet space going and lets other people find it when they need something steady to listen to before sleep or while relaxing after a long day. No pressure, of course, just a small invitation while we get comfortable with the subject. So let's ease into the setting first, because understanding the environment explains why these early animals ended up so unusual. The Paleozoic era began roughly 541 million years ago and lasted about 290 million years. That is a very long stretch of time, far longer than the entire history of mammals, including humans. During most of that era, oceans dominated the planet. Continents existed, but life on land was limited or absent for large portions of that time. The sea was the main stage where biological experimentation happened. Water has properties that encourage certain kinds of growth. It supports weight, distributes nutrients, and allows organisms to float or swim rather than constantly fight gravity the way land animals must. That simple physical fact meant early creatures could grow larger shells, longer bodies, or heavier armour without collapsing under their own mass. Size alone doesn't create monsters, but it sets the stage for them. Another factor was chemistry. Early oceans contained dissolved minerals in concentrations somewhat different from today. Calcium carbonate, silica, and other compounds were readily available. Once organisms evolved ways to use those materials, hard shells, plates, spines, and exoskeletons became common. Hard parts serve several purposes. They protect against predators, provide structural support, and improve mobility in water. Once some organisms developed protective shells, others evolved methods to break those shells. That back-and-forth escalation is sometimes called an evolutionary arms race, and it played a major role in producing animals that look formidable even by modern standards. Temperature and oxygen levels also mattered. Oxygen concentrations in the oceans gradually increased during parts of the Paleozoic. More oxygen supports higher metabolism, which allows animals to move faster, hunt actively, and sustain larger bodies. Low oxygen favours simpler, slower organisms. Higher oxygen opens the door to energetic predators. Food availability was another piece of the puzzle. Microscopic algae and plankton flourished in many ancient seas. These tiny organisms form the base of food chains. Once abundant food existed at the bottom, larger consumers followed. Then predators appeared to eat those consumers. Each step up the chain often produced bigger or more specialised animals. Predation itself is worth emphasizing because it fundamentally changed evolution. Early life forms were mostly simple, soft, and small. Once active predators evolved, survival required defenses. Shells thickened, spines lengthened, sensory organs improved. Some animals burrowed into sediment, others swam faster. Some grew larger simply because size discouraged attack. That pressure created creatures that modern viewers often label monsters, even though they were just animals adapting normally. Vision is another surprisingly practical factor. The development of eyes allowed animals to detect prey and avoid danger more efficiently. Fossil evidence shows that eyes became more complex during the early Paleozoic, especially around the Cambrian period. Once sight entered the evolutionary equation, stealth, speed, camouflage, and armour all gained importance. You begin seeing animals with grasping appendages, compound eyes, streamlined shapes. None of this happened overnight. It took millions of years, but the direction becomes clear when fossils are arranged chronologically. Currents and geography contributed too. Ancient continents sat in different positions, shaping ocean circulation. Warm, shallow seas spread across large continental shelves. Those shallow regions often host high biodiversity because sunlight reaches the bottom and nutrients accumulate easily. That environment supported dense ecosystems where competition was intense. It's also helpful to adjust expectations about what counts as a monster. Some Paleozoic predators measured less than a metre long, but relative to the tiny organisms around them, they were dominant hunters. Others grew several metres long, which is impressive even today. Scale matters, context matters. Take arthropods, for example, the group that includes insects, spiders, and crustaceans today. Many early Paleozoic arthropods were marine, and some reached sizes that modern relatives rarely achieve. Exoskeletons provided strength, while water buoyancy reduced structural stress. Some developed large grasping limbs for capturing prey. Others had wide swimming flaps. These designs look unusual now, but they worked effectively at the time. Fish eventually joined the scene as well. Early fish lacked jaws, but later forms developed them, allowing stronger bites and more diverse feeding strategies. Jaws are a major evolutionary innovation. They transform passive filter feeders into active predators. Once jaws appeared, ecological dynamics shifted again. Reefs also played a role. Reef structures provided habitat complexity, hiding places, and concentrated food sources. Wherever reefs form, biodiversity increases. That leads to specialization, competition, and occasionally large predators that exploit crowded ecosystems. Mass extinctions punctuated the Paleozoic, periodically resetting ecosystems. Volcanic activity, climate shifts, ocean chemistry changes, and asteroid impacts all contributed at different times. After each extinction, surviving organisms diversified rapidly into newly available niches. Those recovery periods sometimes produced unusual forms because evolutionary pathways reopened. Fossil preservation influences what we know. Hard shells fossilize more easily than soft tissue, so armoured creatures appear prominently in the record. That can exaggerate how monstrous the fauna seems. Soft-bodied animals certainly existed but left fewer traces. Scientists use rare deposits with exceptional preservation to fill gaps. One subtle point often overlooked is timescale perception. When we look at a fossil montage in a documentary, millions of years may pass in seconds. In reality, each anatomical change took countless generations. No sudden appearance of monsters occurred. Gradual accumulation produced them. Climate stability during certain intervals allowed ecosystems to mature. Stable environments let evolutionary arms races continue without constant disruption. That stability can encourage size increases and specialization. Then there is simple ecological opportunity. Early in complex life history, many ecological roles were unoccupied. The first organisms to exploit them faced little competition. That freedom encourages experimentation in body plans. You may notice something reassuring here. None of these processes are mysterious. Physics, chemistry, biology, and time are enough. No hidden forces required, just consistent natural rules operating over immense durations, and yet the results look dramatic from our perspective, because modern oceans have different balances. Marine mammals dominate large predator roles today, whereas Paleozoic seas relied heavily on arthropods and armoured fish. Seeing unfamiliar dominance patterns creates that monster impression. Another grounding observation involves sensory perception. Modern humans instinctively react strongly to unfamiliar body shapes. Large compound eyes, segmented limbs, armoured plates. Those features trigger caution because they differ from mammals we recognise easily. That reaction shapes how we describe ancient animals. Sediment layers tell detailed environmental stories. Grain size, chemical composition, fossil distribution all help reconstruct habitats. That evidence shows many Paleozoic oceans were nutrient-rich but also periodically unstable. Fluctuating oxygen and temperature levels likely encouraged adaptable, resilient organisms. Resilience often correlates with protective anatomy. Armour, burrowing behaviour, and flexible diets all increase survival odds. Over time, those traits accumulate. Even reproduction strategies matter. Species producing many offspring can adapt faster. Rapid reproduction supports evolutionary experimentation. Ocean depth gradients created multiple ecological zones: shallow coastal waters, mid-depth shelves, deep basins. Each zone hosted different organisms, sometimes connected by migration. Chemical signalling between organisms also evolved early. Predators detect prey chemically. Prey detect predators. That invisible communication shapes behaviour and anatomy. Movement efficiency became important as predation intensified. Streamlined bodies, fins, jointed limbs all improve mobility. Some creatures specialised in ambush tactics, others in pursuit. Noise likely played a role too. Water transmits vibration well. Detecting pressure waves helps locate movement even without vision. As all these factors combined, ecosystems grew increasingly complex. Food webs expanded, interdependencies formed, and complexity tends to produce standout species simply because variation increases. By this point, the setting begins to feel less alien and more understandable. Ancient seas were not chaotic fantasy environments. They followed logical biological patterns. The reason those environments produced Earth's earliest so-called monsters becomes clearer. Supportive buoyancy, abundant minerals, rising oxygen, evolving predation, expanding ecological niches, and immense time. That combination reliably generates large, specialised animals. From here, it becomes useful to look more closely at what those oceans were actually like day to day, what conditions organisms experienced continuously, because the environment itself shaped every adaptation that followed. And easing into that broader environmental picture helps make sense of how the earliest marine ecosystems set the stage for everything that came next. If you imagine the Paleozoic ocean before large predators became common, it helps to clear away the dramatic imagery that often comes with prehistoric documentaries. No constant battles, no oversized hunters dominating every scene, and certainly no cinematic tension. For very long stretches of time, those ancient seas were comparatively quiet places biologically speaking. Activity existed, of course, but it was mostly subtle. Tiny organisms drifting, simple animals feeding slowly along the seafloor, chemical processes unfolding steadily. Life was present, but it had not yet escalated into the visible predator-prey dynamics that later defined marine ecosystems. The ocean itself looked different from what you would see standing on a modern coastline. Continents were arranged in unfamiliar positions, many clustered closer together or partially submerged. Large shallow seas spread across continental interiors, something rare today. These shallow marine environments are particularly important because sunlight penetrates easily, encouraging photosynthesis. That leads to plankton blooms, which then support larger organisms. So even before obvious monsters appeared, the stage was already being built through energy flow and nutrient cycling. Water chemistry deserves attention because it shaped early life directly. Paleozoic oceans contained higher levels of certain dissolved minerals, especially calcium and magnesium compounds. When early organisms evolved the ability to incorporate these minerals into shells or skeletons, it created structural advantages. But initially, most life forms were soft-bodied. Hard parts came gradually. Before that shift, preservation in the fossil record was limited, which sometimes makes the earliest oceans seem emptier than they actually were. Temperature patterns were also somewhat different. Global climates during early Paleozoic periods tended to be warmer overall, with less pronounced polar ice than in later eras. Warm water holds slightly less dissolved oxygen than cold water, but circulation patterns and atmospheric composition compensated in various ways. Ocean mixing transported oxygen downward, supporting animal metabolism even before complex predators appeared. Currents played a stabilizing role. Large-scale circulation distributes heat, nutrients, and dissolved gases. These flows existed in the Paleozoic just as they do now, though continental placement altered specific routes. Stable circulation reduces environmental shocks, which helps ecosystems develop continuity. Continuity, in turn, allows evolutionary experimentation to accumulate gradually, rather than being repeatedly reset. Sunlight was essential, not just for warmth, but for energy input. Photosynthetic microorganisms formed extensive populations near the surface. Cyanobacteria, algae, and early plankton converted solar energy into chemical energy. Everything else in the ecosystem ultimately depended on that process. Even large predators millions of years later trace their energy lineage back to those microscopic producers. Sediment composition offers another practical clue about these environments. Fine muds, silts, and carbonate deposits accumulated on sea floors. These sediments preserved burrows, feeding traces, and microbial mats. Before large animals stirred the sediment extensively, seafloor layers often remained stable for long periods. That stability allowed microbial communities to dominate certain habitats. Microbial mats are particularly important. They form dense layers of bacteria and algae on sediment surfaces. Some early animals grazed on these mats, others lived within them. These mats also influenced oxygen distribution, because photosynthesis produces oxygen, while decomposition consumes it. The balance between those processes shaped local conditions. Noise levels in these oceans were likely lower than today. Modern seas contain ship traffic, industrial sound, and dense populations of marine mammals producing vocalizations. Paleozoic oceans lacked those contributors. Biological noise existed, but overall acoustic environments were probably simpler. That influences sensory evolution, because organisms adapt to the signals present in their surroundings. Another factor involves nutrient delivery from land. Early Paleozoic landscapes lacked extensive vegetation. Soil formation was limited compared to modern conditions. Rivers still transported minerals into the sea, but the pattern differed. As plants later colonized land, nutrient runoff increased, affecting ocean productivity. So before that terrestrial expansion, marine ecosystems operated under slightly different nutrient dynamics. Volcanic activity contributed intermittently. Underwater eruptions released minerals and heat locally. Hydrothermal vents existed, providing chemical energy independent of sunlight. Even today, vent ecosystems support specialized organisms. In the Paleozoic, similar environments likely hosted early complex life forms, though fossil evidence remains limited due to preservation challenges. Salinity levels fluctuated regionally. Enclosed shallow seas sometimes experienced evaporation, concentrating salts. Open ocean areas remained more stable. Organisms adapted accordingly. Some tolerated wide salinity ranges, others specialized narrowly. Storm patterns influenced sediment disturbance. Without extensive coastal vegetation, Shorelines were more exposed. Storm-driven waves redistributed sediments, periodically altering habitats. These disturbances prevented ecological stagnation, but also created opportunities for colonization. Oxygen levels in the atmosphere gradually rose through earlier geological periods, and by the Paleozoic they supported increasingly active animals. However, oxygen distribution in the ocean was uneven. Some deeper basins experienced low oxygen conditions, limiting animal diversity there. Shallow, well-mixed waters remained more hospitable. The absence of large land predators also mattered indirectly. Nutrient cycles linking land and sea were simpler. Marine ecosystems evolved somewhat independently at first. Later terrestrial ecosystems fed back into ocean chemistry more strongly. Reproduction strategies among early marine animals tended toward high output. Many organisms released large numbers of eggs or spores into the water. This approach increases survival odds in environments where predation is initially low, but environmental unpredictability remains. Locomotion among early animals was generally slow. Many were sessile, meaning fixed in place, or capable only of limited crawling. Energy efficiency favoured minimal movement. Without active predators, speed provided less advantage. Feeding strategies were correspondingly straightforward. Filter feeding dominated. Organisms extracted suspended particles from water. Grazing on microbial mats was also common. Complex hunting behaviours emerged later. Body symmetry patterns began diversifying during this time. Bilateral symmetry, allowing directional movement, became more common. That shift eventually supported active locomotion. Sensory organs were initially simple. Chemical sensing likely preceded vision in complexity. Detecting dissolved substances helps locate food or avoid harmful conditions. Water clarity varied widely. Plankton blooms reduce visibility, while clear nutrient-poor waters increase it. Visual systems evolve differently depending on clarity. So early ocean optics influenced eye development trajectories. It's useful to remember that evolutionary innovation rarely happens uniformly across the globe. Different basins, climates, and chemical conditions produced regional variations. Some areas saw rapid diversification earlier than others. Plate tectonics constantly reshaped ocean basins. Seafloor spreading created new habitats. Subduction zones recycled crust. These geological processes influenced ocean depth, circulation, and chemistry continuously. Carbon cycling deserves a mention because it ties climate and biology together. Marine organisms incorporating carbon into shells remove carbon dioxide from the water. Over long periods, that influences atmospheric composition. Feedback loops between life and climate were already operating. Another subtle but significant factor was ultraviolet radiation. Early ozone levels were still stabilizing. Water absorbs UV efficiently, so marine environments provided protection. That likely encouraged continued ocean habitation before land colonization intensified. Ecological niches expanded gradually. Once one species exploited a resource successfully, others evolved variations. Diversity builds incrementally. Competition remained relatively low at first simply because complex life had not diversified extensively yet. Low competition allows experimentation. Eventually, competition intensifies, encouraging specialization. Chemical communication among microorganisms may have influenced multicellular evolution. Signaling molecules coordinate behavior even in simple colonies. Tidal patterns influenced coastal ecosystems. Tides expose and submerge shallow habitats regularly, encouraging adaptability. Glaciation events occurred intermittently later in the Paleozoic. Ice formation alters sea levels, temperature gradients, and salinity. Those shifts influence marine biodiversity cycles. Sediment oxygenation improved as burrowing animals evolved. This bioturbation mixes sediments, affecting microbial communities and nutrient availability. Predator absence doesn't mean complete safety. Environmental stresses like temperature swings, salinity shifts, and oxygen fluctuations still posed risks. Population densities fluctuated seasonally and regionally. Plankton cycles influenced everything above them. Symbiotic relationships began appearing. Organisms benefiting mutually from proximity gained survival advantages. Metabolic diversity expanded. Some organisms relied on oxygen, others tolerated low oxygen conditions. Chemical defenses evolved even before physical defenses. Toxins discourage predation once predators appear. Structural proteins like collagen enabled stronger tissues, paving the way for more complex bodies. As these developments accumulated, ecosystems gained layers of interaction. Complexity breeds opportunity for larger, more capable animals. That gradual build-up leads naturally toward the appearance of organisms we recognize as dominant predators. But understanding the baseline conditions first keeps the story grounded, because before any creature could become imposing in size or behavior, the ocean itself had to become a stable, resource-rich environment, capable of sustaining increasingly sophisticated forms of life. And that environmental groundwork quietly shaped everything that followed. Long before anything that could reasonably be called a monster moved through the water, the planet itself was still settling into habits. Continents drifted slowly, oceans opened and closed, volcanic gases seeped into the atmosphere, and the surface of Earth was shaped more by geology than biology. If you could somehow stand on a shoreline during the early Paleozoic, you would not hear crashing cities or distant aircraft. You would hear wind, waves, and perhaps the subtle crackle of shifting sediment. Beneath the surface, most activity was quiet and dispersed. Life existed, but it did not yet dominate the character of the world. The phrase deep water silence is not poetic exaggeration. Compared to modern oceans, the early Paleozoic seas likely felt biologically subdued in many regions. There were no whales vocalizing across kilometers of water, no schools of large fish flashing in coordinated motion, no coral reefs buzzing with snapping shrimp. Instead, vast areas of seafloor were covered with fine sediment, microbial mats, and scattered simple animals moving slowly or remaining anchored in place. The planet itself was younger in a geological sense. Not newly formed, of course, but still early in the long arc of complex life. Oxygen levels in the atmosphere had risen significantly compared to the earliest eras, yet they had not stabilized at modern concentrations. The chemistry of both air and sea continued adjusting to biological and tectonic processes. Ocean basins during this time were shaped by active plate tectonics. Seafloor spreading created new crust along mid-ocean ridges. Subduction zones consumed old crust elsewhere. These processes continuously recycled material between Earth's surface and interior. Every ridge and trench influenced water circulation patterns. Heat from the mantle warmed surrounding waters locally, contributing to chemical gradients that certain microorganisms exploited. Continental positions were unfamiliar. Large land masses clustered differently, and sea levels fluctuated frequently. At times, shallow epicontinental seas flooded broad areas of continental crust. These shallow seas were crucial nurseries for evolving life because sunlight reached the bottom and nutrients accumulated efficiently. Yet even there, early ecosystems lacked the dramatic scale of later periods. The deep ocean floors were darker and more chemically variable. Oxygen did not always penetrate to great depths. In some basins, low oxygen conditions limited animal diversity. Where oxygen was scarce, only organisms adapted to such environments could survive. That constraint reduced complexity in those regions. It's helpful to consider how sound travels underwater. Water transmits vibrations efficiently, but in the absence of large mobile animals, acoustic environments would have been dominated by natural physical processes, current-shifting sediment, distant underwater landslides, perhaps volcanic rumblings. Biological noise would have been subtle. Small arthropods scraping across sediment, filter feeders moving water across feeding structures. Nothing like the layered chorus of modern marine life. Sunlight filtered into upper layers predictably each day. Diurnal cycles existed long before complex animals. Photosynthetic microorganisms responded to light patterns, adjusting metabolism accordingly. That rhythm created daily fluctuations in oxygen production near the surface. These changes influenced local chemistry and shaped microhabitats. Temperature gradients were relatively stable in many regions. Warm surface waters overlay cooler, deeper layers. Stratification limited vertical mixing in some basins, contributing to oxygen-poor depths. In shallower seas, wind-driven mixing distributed nutrients and gases more evenly. Sediment accumulated gradually on sea floors. Fine particles settled from suspension. Without large burrowing animals initially to disturb layers, these sediments often remained laminated and undisturbed. Such undisturbed layers preserved chemical signatures and occasional impressions of soft-bodied organisms. Life at this stage was diverse but mostly small. Early arthropods, worms, simple mollusk-like forms, and various enigmatic organisms occupied niches. Many fed by filtering microscopic particles from water. Others grazed slowly across microbial mats. Movement tended to be limited because energy budgets were constrained and predation pressure was still developing. The absence of intense predation is key. In ecosystems where few active hunters exist, there is less immediate pressure to develop heavy armour, rapid escape mechanisms, or complex sensory systems. That does not mean evolution stops. It simply proceeds along different lines, emphasizing efficiency and reproduction over defense and aggression. Water chemistry still bore traces of earlier geological epochs. Mineral availability varied regionally. Organisms experimenting with mineralized structures began appearing, but these innovations were not yet universal. Some lineages developed simple shells or spicules, while others remained soft-bodied. Carbon cycling between ocean and atmosphere continued adjusting. Photosynthesis removed carbon dioxide from water, while volcanic emissions added it. Over long periods, these processes influenced global climate. During stable intervals, ecosystems matured quietly. It's worth remembering that evolutionary innovation often happens in modest steps. The first hint of a new feeding structure, a slightly more sensitive sensory patch, a marginally stronger body wall. No sudden transformation into formidable forms, just gradual improvement. Storm events periodically disturbed shallow environments. Strong waves redistributed sediment and could temporarily displace organisms. However, these disturbances also created open space for colonization. In ecosystems with low competition, open space encourages diversification. The seafloor itself presented varied microhabitats. Ripple marks formed by currents created ridges and troughs. Small depressions collected organic matter. Microbial mats stabilized sediment surfaces in some areas, while other regions remained loose and shifting. Chemical gradients near hydrothermal vents supported unique communities. Even in the absence of sunlight, life exploited energy from chemical reactions. These communities likely remained isolated from shallow water ecosystems, but contributed to overall diversity. Tidal forces influenced coastal areas. Twice daily exposure and submergence in some regions required adaptability. Organisms capable of tolerating fluctuating moisture levels had advantages near shorelines. Because land plants were not yet widespread, terrestrial runoff differed from modern patterns. Rivers carried minerals but less organic debris. That influenced nutrient composition in coastal waters. Reproductive strategies favoured wide dispersal. Many organisms released gametes into open water, relying on currents for distribution. This method promotes gene flow across distances, but also produces high mortality rates. In the absence of dominant predators, body plans diversified in shape and symmetry. Bilateral forms capable of directional movement became more common, laying groundwork for later active locomotion. Energy flow through food webs remained relatively simple. Primary producers at the base, filter feeders and grazers above them. Few specialized hunters yet shaped those webs. It is important not to imagine emptiness. Even without dramatic creatures, these oceans were biologically active. They simply lacked the ecological intensity that would later define them. Atmospheric conditions above influenced everything below. Wind patterns affected surface mixing. Solar radiation levels influenced productivity. As plate tectonics repositioned continents gradually, ocean currents shifted. Climate zones migrated. These slow geological adjustments provided varied selective pressures over millions of years. Genetic mutations accumulated steadily within populations. Natural selection filtered those variations based on environmental fit. Most changes were minor, but occasionally they opened new possibilities. Over time, subtle interactions increased. A slightly faster grazer encounters more food. A slightly tougher shell resists minor damage. A marginally improved sensory organ detects chemical traces earlier. Each improvement sets the stage for further adaptation. Complexity does not require drama. It requires stability and variation over long intervals. The early Paleozoic oceans provided exactly that combination. Gradually, the ecological balance began shifting. As certain organisms evolved more effective feeding mechanisms, pressure on prey increased. Once even a modest predator gains efficiency, prey populations respond. The first active hunters likely targeted soft-bodied organisms. Prey that survived reproduced. Defensive traits spread. These dynamics unfolded quietly at first. Visual systems began improving. Light detection evolved into rudimentary image formation. Once sight becomes reliable, movement gains advantage. Arthropods in particular showed capacity for morphological innovation. Segmented bodies allow specialization of appendages. That modular design encourages experimentation. Still, much of the ocean floor remained tranquil compared to later periods. Vast areas supported slow, steady ecological processes. Oxygen levels gradually increased in certain intervals, supporting higher metabolism. Higher metabolism supports faster movement and more complex behavior. Climate oscillations occasionally interrupted stability. Cooling or warming episodes altered habitats. Species adapted or disappeared accordingly. Despite these changes, the overall character of early Paleozoic seas was one of gradual build-up rather than immediate escalation. The silence, if you imagine it, was not absence but anticipation. Biological systems were assembling components that would later combine into more visibly dominant forms. Every mineralized plate, every improved sensory structure, every incremental metabolic boost added to a growing toolkit. Eventually that toolkit would enable animals capable of active pursuit, defense, and impressive size relative to their environment. But before those forms fully emerged, the planet spent millions of years refining conditions quietly, building complexity layer by layer beneath waves that looked ordinary from the surface, and understanding that extended preparation makes it easier to see how the next phase unfolded when evolutionary interactions intensified and the first unmistakably imposing marine creatures began to move through ecosystems that had been patiently assembling the capacity to sustain them. If you picture the long build-up we've been talking about, slow environmental preparation, small organisms experimenting with simple body plans, relatively quiet sea floors, then the next development feels abrupt by comparison, although it still unfolded over millions of years. What scientists call the Cambrian explosion marks a period roughly beginning around 541 million years ago, when the fossil record starts showing a noticeable increase in the diversity, size, and structural complexity of marine animals. The word explosion can be misleading because nothing happened instantly. It was rapid only on geological timescales. Still, compared with earlier eras, the change was dramatic enough that it stands out clearly in sediment layers across multiple continents. Before this interval, most known organisms were either microscopic, soft-bodied, or structurally simple. Afterward, you start seeing creatures with defined heads, segmented bodies, jointed limbs, compound eyes, hard shells, spines, and increasingly specialized feeding mechanisms. That shift matters because complexity allows new ecological roles. Once animals can see better, move efficiently, grasp prey, or defend themselves physically, interactions intensify. Ecosystems become layered and dynamic rather than quiet and uniform. Several factors likely contributed to this increase in complexity, and none works alone. Oxygen availability is one of the most discussed. Evidence from geochemical signatures suggests oxygen levels rose in both atmosphere and shallow oceans during the late Precambrian and early Cambrian periods. Oxygen supports higher metabolic rates. Higher metabolism enables sustained movement, active hunting, and development of energy-intensive organs like eyes and nervous systems. Without sufficient oxygen, complex bodies struggle to function effectively. Genetic developments also played a major role. Many biologists point to the expansion and diversification of developmental regulatory genes, particularly Hox genes. These genes help determine body layout during embryonic development. When regulatory gene networks diversify, organisms gain flexibility in body design. Limbs, segments, sensory structures, and specialized tissues become easier to evolve because developmental instructions can vary without disrupting overall viability. Genetic complexity does not guarantee visible anatomical change, but it provides the toolkit. Environmental stability likely contributed as well. Periods of relative climate stability allow evolutionary experimentation to accumulate. If conditions change too quickly, populations cannot adapt fast enough. During the early Cambrian, certain marine environments appear to have remained stable long enough for diversification to flourish. Predation pressure is another key element. Once even moderately effective predators evolved, prey species faced new survival challenges. Defense strategies followed. Shells thickened, spines appeared, burrowing behavior increased. Some organisms developed faster movement. Predator-prey feedback loops accelerate evolutionary change because each adaptation in one group creates pressure on another group to respond. One of the most striking features in Cambrian fossils is the appearance of mineralized skeletons. Calcium carbonate shells, silica-based spicules, phosphate plates. These structures fossilize well, which partly explains why the fossil record suddenly seems richer. It is not just that more animals existed, more of them left durable remains. That preservation bias amplifies the apparent abruptness of the diversification. Shallow marine environments played a central role. Sunlight penetrates these waters, supporting photosynthetic plankton. Nutrients delivered from weathering continents accumulated there. These conditions foster high productivity. High productivity supports complex food webs. Many classic Cambrian fossil sites represent shallow continental shelf environments. The Burgess Shale in Canada and the Chengjiang deposits in China provide particularly detailed windows into this time. Exceptional preservation in fine sediments captured soft tissues, digestive systems, eyes and appendages rarely fossilized elsewhere. These sites reveal animals that would otherwise remain unknown. Some look surprisingly modern in body organization. Others appear unfamiliar even now. Among these organisms are early arthropods, including forms related to trilobites. Arthropods possess segmented bodies, jointed limbs, and exoskeletons. This modular construction allows specialization. Some appendages become legs, others feeding structures, others sensory organs. That flexibility encourages diversification. Early mollusk-like animals also appear, along with brachiopods, echinoderms in primitive forms, sponges, and various worm-like organisms. Many major animal phylo-recognizable today trace origins back to this period. That continuity is important. The Cambrian did not produce isolated curiosities that later disappeared entirely. It established foundational lineages. Eyes deserve special attention because they dramatically alter ecological dynamics. Fossils show complex compound eyes in some Cambrian arthropods. Vision enables active hunting, navigation, and social interactions. Once sight evolves, camouflage, speed, and defensive armor become advantageous. Sediment disturbance increased as animals burrowed more actively. This process, called bioturbation, mixes oxygen into sediments and alters microbial communities. Bioturbation changes nutrient cycling and creates new ecological niches. It represents an ecosystem engineering effect driven by animal behavior. Food webs became more intricate. Instead of simple producer-consumer chains, multiple trophic levels developed. Detritivores processed organic debris. Grazers fed on microbial mats. Predators hunted grazers. Scavengers recycled remains. Complexity increased resilience, but also competition. Competition itself encourages specialization. If multiple species exploit similar resources, those that diversify slightly often gain advantage. Over time, niches subdivide. Specialized feeding structures, sensory adaptations, and reproductive strategies emerge. Chemical signaling also evolved further. Detecting dissolved molecules helps locate food or avoid danger. Some organisms likely released deterrent chemicals to discourage predation. Others evolved receptors to detect those signals. Hydrodynamics influenced body shapes. Efficient swimming reduces energy expenditure. Streamlined forms appear in some Cambrian organisms. Others specialized for crawling or burrowing depending on habitat. Reproductive diversity expanded. Some species produced numerous small offspring. Others invested more resources in fewer, larger offspring. Each strategy has advantages depending on environmental stability and predation pressure. Climate fluctuation still occurred, but overall marine productivity remained sufficient to sustain diversification. Continental configurations influenced current patterns, which distributed nutrients unevenly across basins. Carbon cycling accelerated as organisms incorporated carbon into shells. Burial of these shells in sediments affects atmospheric carbon dioxide over geological timescales, linking biological evolution to climate regulation. It is tempting to interpret the Cambrian explosion as a singular turning point, but it is better understood as part of a continuum. Preceding periods laid groundwork. Subsequent periods refined and expanded complexity. Importantly, not all Cambrian animals were large. Many remained small. Size variation increased, but miniature organisms still dominated numerically. Large size simply becomes more visible in fossils and reconstructions. Ecological interactions intensified gradually. The earliest predators may not have been dramatically efficient. Even moderate predation can reshape ecosystems over time. Some Cambrian animals possessed unusual anatomies not easily classified within modern groups. These forms illustrate evolutionary experimentation. Most lineages eventually disappeared, but they demonstrate the range of possibilities explored. Nervous systems became more centralized in certain groups. Concentrated nerve tissue improves coordination and sensory processing. That supports active behavior. Muscle development advanced. Efficient musculature enables sustained movement. Coupled with improved oxygen supply, this allowed animals to explore habitats more actively. Exoskeleton molting appears in arthropods. Periodic shedding allows growth despite rigid outer coverings. Molting cycles introduce vulnerability periods but enable size increase. Sediment color changes in fossil layers sometimes indicate oxygenation shifts associated with increased biological activity. These geochemical signals corroborate fossil evidence. Stable isotope analysis provides clues about ancient diets and environmental conditions. Ratios of carbon and oxygen isotopes reflect metabolic processes and water temperatures. Trace fossils, such as burrows and feeding marks, complement body fossils. They reveal behavior even when bodies are not preserved. All these lines of evidence converge on a picture of escalating biological complexity during the Cambrian. Not sudden appearance from nothing, but rapid diversification relative to previous periods. From an ecological standpoint, the emergence of structured food webs marks a critical shift. Once multiple trophic levels exist, ecosystems gain depth and feedback loops. This depth allows certain organisms to become dominant in their niches. Dominance often correlates with traits that humans interpret as monstrous: size, armour, specialised predatory appendages. However, at this stage, truly gigantic forms remained rare. The groundwork was still being laid. Future periods would push size and specialisation further. Understanding this phase clarifies that complexity does not arise randomly. It follows environmental opportunity, genetic capability, and ecological interaction. The Cambrian period provided those conditions simultaneously. Oxygen increase, genetic flexibility, stable shallow seas, expanding food webs, emerging predation. Each factor alone would have limited effect. Together they accelerated diversification noticeably. From here, the narrative naturally moves towards specific organisms that exemplify these changes, creatures whose anatomy reflects the new ecological pressures directly, and examining those animals more closely makes it easier to see how evolutionary experimentation translated into the first marine predators that begin to look formidable even by modern standards. As complexity continued building in those early Paleozoic seas, one development began changing marine ecosystems in a very practical way. Predators were no longer just small opportunists picking at microbial mats or drifting plankton. Some animals were starting to specialise in actively hunting other animals. That shift marks a clear ecological turning point, because once efficient hunters appear, everything else adapts in response. One of the clearest examples from the Cambrian period is a creature called Anomalocaris, an animal that for a long time puzzled scientists because its body parts were originally discovered separately and misidentified as entirely different organisms. Early fossil finds included a circular mouth structure, grasping appendages, and a segmented body with lateral swimming flaps. At first, paleontologists thought these pieces belonged to unrelated species. Only later, particularly after discoveries in exceptionally preserved Cambrian deposits like the Burgess Shale, did researchers realise they were parts of one large predator. That realisation immediately changed interpretations of Cambrian ecosystems, because it demonstrated that sizable mobile hunters already existed. Compared with many animals of its time, Anomalocaris was large. Estimates vary depending on species, but some individuals reached lengths approaching 1 metre or slightly more. That may not sound enormous today, yet in an ecosystem where many organisms were only a few centimetres long, it represented a dominant presence. Size alone provides advantages in predation. Larger animals can capture a wider range of prey, travel farther efficiently, and resist predation themselves. Its anatomy reveals several important evolutionary developments. The frontal appendages are perhaps the most distinctive feature. These were flexible, segmented limbs positioned near the head, lined with spines that likely helped grasp prey. Unlike simple feeding tentacles seen in earlier organisms, these appendages appear adapted for active capture. Jointed movement allows precise control. That suggests coordination between sensory input and motor response, an indication of increasingly complex nervous systems. The mouth structure is also notable. Often described as pineapple slice-shaped, it consisted of overlapping plates forming a circular opening. This design likely allowed crushing or gripping prey rather than simple suction feeding. Debate continues about exactly how it functioned, but wear patterns on some fossils indicate mechanical stress consistent with predation. Locomotion provides further clues. Rows of lateral flaps along the body likely generated propulsion through undulating motion. Modern analogies include certain cephalopods or rays using fins for efficient swimming. This propulsion system would have allowed relatively rapid, energy-efficient movement through water, supporting active hunting rather than passive drifting. Vision appears to have been advanced for the time. Fossils show large compound eyes in some species attributed to this group. Compound eyes provide wide fields of view and sensitivity to motion. Detecting moving prey or predators becomes easier with such visual capability. Once vision reaches this level, ecological interactions change significantly. Camouflage, speed, and defensive structures gain importance. The emergence of a predator like Anomalocaris likely influenced prey evolution immediately. Trilobites, early arthropods abundant in Cambrian seas, show evidence of defensive adaptations including enrollment behaviour, thicker exoskeletons and spines in some lineages. Enrollment allows a trilobite to curl into a protective ball, shielding vulnerable soft parts. That behaviour makes sense in an environment with predators capable of grasping exposed tissue. There is ongoing discussion about whether Anomalocaris could crush hard shells effectively. Some trilobite fossils show damage patterns possibly consistent with predatory attacks, though not all researchers agree on the interpretation. Regardless, the presence of even moderately effective predators encourages defensive innovation. Predation pressure also influences behaviour. Burrowing becomes more common as prey species seek refuge in sediment. Increased burrowing activity oxygenates sediment layers, altering microbial communities and nutrient cycling. So a predator does not just affect its prey, it reshapes entire ecosystems indirectly. Another aspect involves energy flow. Active predators require substantial caloric intake. That requirement encourages productive lower trophic levels. Plankton populations, grazers, and intermediate consumers all contribute to sustaining apex hunters. A stable energy pyramid begins forming. Fossil distribution suggests Anomalocaris and related forms occupied multiple marine environments, particularly shallow continental shelves where biodiversity was highest. Shallow waters support abundant prey and favourable temperatures for metabolism. Hydrodynamics of the body also indicate adaptation to open water movement rather than strictly bottom-dwelling habits. That expands ecological reach. Mobile predators connect previously separate ecological zones by moving between them. Reproductive strategies for such animals remain uncertain, because soft tissues rarely fossilize, but high offspring numbers are plausible. Many marine arthropod-like organisms use broadcast spawning. Large numbers of larvae increase survival probability despite predation. Growth through molting, if present as in many arthropods, would introduce vulnerable phases. Predators themselves face risks during molting. That dynamic contributes to complex predator-prey cycles. The presence of apex predators often stabilizes ecosystems by controlling population sizes of intermediate consumers. Without predators, certain prey species might overexploit resources. With predators, balance emerges. Chemical sensing likely supplemented vision. Detecting dissolved organic compounds helps locate prey even in low visibility conditions. Combined sensory modalities improve hunting success. Muscle attachment structures inferred from fossil morphology suggest reasonably strong musculature. Efficient muscle systems enable sustained swimming and rapid bursts when capturing prey. Comparisons with later Paleozoic predators show evolutionary continuity. Jointed appendages, sensory specialization, and active locomotion remain successful strategies for hundreds of millions of years. It is worth emphasizing that labeling such animals monsters reflects human perception rather than biological reality. They were simply effective predators adapted to their environment. Their unfamiliar appearance contributes to the dramatic impression. The rise of predators like Anomalocaris coincides with broader ecological diversification. Reef-like structures expanded, filter feeders diversified, detritus processing increased, ecosystems gained depth and resilience. Competition among predators also likely existed. Multiple hunting species targeting similar prey encourages niche differentiation. Slight differences in hunting method, habitat preference, or diet reduce direct competition. Climate conditions during parts of the Cambrian supported relatively stable marine productivity. Stable productivity allows predator populations to persist without frequent collapse. Fossil evidence sometimes includes gut contents, providing direct insight into diet. Some specimens show remains interpreted as small arthropods or soft-bodied organisms. These findings support active predation hypotheses. Biomechanical modelling helps estimate bite forces and swimming efficiency. Computer simulation suggests reasonable predatory capability. though uncertainties remain due to incomplete fossil data. The broader significance of these predators extends beyond their own lineage. Once active hunting becomes common, evolutionary pace accelerates. Defense, speed, sensory development, and ecological specialization all intensify. Sediment records following this period show increased trace fossil diversity. More complex burrows, feeding marks, and movement patterns appear. That behavioral diversification reflects ecological pressure from predators. From a physiological standpoint, sustained swimming requires efficient oxygen transport systems. While detailed soft tissue is rarely preserved, anatomical features imply circulatory adaptations adequate for active lifestyles. Some Cambrian predators may have exhibited diel activity patterns, feeding at certain times of day depending on prey availability. Light cycles influence marine behavior even today. Taphonomic processes, meaning how organisms become fossilized, sometimes favor preservation of larger animals. That can exaggerate perceived dominance. Smaller organisms remained abundant but less visible in the record. Interactions between predators and early reef builders likely shaped reef evolution. Predation pressure influences reef organism morphology and community structure. The evolutionary success of arthropod body plans is evident here. Segmentation, jointed limbs, and exoskeletons remain effective across multiple geological eras. Genetic diversity within predator populations promotes adaptability. Environmental fluctuations select for flexible traits. Ocean circulation patterns distribute larvae widely, promoting gene flow between populations. That connectivity reduces extinction risk. As ecosystems continued developing, additional predator groups emerged. Jawed fish would eventually join the scene, introducing new predatory strategies. Each new predator type layered additional pressure on prey species, driving further diversification. This gradual escalation illustrates how complex ecosystems arise from simple beginnings through cumulative interaction. Looking at this phase in isolation can make it seem dramatic, but within the broader Paleozoic timeline, it represents one step in a continuing process. Understanding these early apex predators clarifies why later oceans hosted even larger and more specialized hunters. The ecological groundwork was already established. From here, examining defensive responses among prey species becomes particularly informative, because armor, spines, and protective behaviors expanded significantly once active predators became a consistent environmental factor. And those defensive innovations help explain how marine life continued diversifying under increasing ecological pressure. Once active predators became a consistent feature of Paleozoic oceans, the balance of marine ecosystems began shifting in a very practical way. Earlier, survival often depended mainly on environmental tolerance, efficient feeding, and reproduction. Now, avoiding being eaten became an equally important concern. That change pushed many organisms towards structural defenses. Armor, spikes, shells, thicker exoskeletons, and behavioral adaptations all emerged gradually, not as sudden inventions, but as incremental responses to steady predation pressure. It helps to think of this process as an evolutionary feedback loop rather than a one-time event. Predators improve hunting ability, prey evolve defenses, predators adapt again, and the cycle continues. Over millions of years, this feedback can produce animals that look unusually fortified compared with earlier life forms. The Paleozoic oceans provide some of the earliest clear examples of this arms race in action. One of the most obvious defensive innovations was biomineralization, the biological use of minerals to create hard external structures. Calcium carbonate became particularly common. Organisms capable of depositing this mineral into shells or plates gained immediate advantages. Hard surfaces resist crushing, biting, and tearing. Even partial mineralization can discourage predation by increasing handling time. If a predator spends too much energy attempting to consume a particular prey item, it may move on to easier targets. That simple cost-benefit calculation influences survival outcomes strongly. Trilobites illustrate this process well. Early trilobites already possessed exoskeletons, but later species often show thicker shells, more pronounced spines, and the ability to enroll tightly. Enrollment allows the animal to curl its segmented body so that only hardened surfaces face outward. Fossil evidence includes specimens preserved in enrolled positions, suggesting defensive behaviour was routine rather than occasional. Spines represent another effective deterrent. Spines increase apparent body size, make swallowing difficult, and can cause physical injury to predators. Some Paleozoic arthropods and mollusk-like organisms developed elaborate spine arrangements. These structures do require metabolic investment, so their persistence indicates clear survival advantages. Shell thickening occurred in multiple unrelated groups, an example of convergent evolution. Brachiopods, mollusks, and certain echinoderms all show increased shell robustness over time. Independent development of similar defences suggests strong selective pressure across ecosystems. Sediment burrowing also became more common. Instead of relying solely on external armour, some organisms escape predation by living partially or fully within sediment. Burrows provide physical concealment and sometimes chemical protection. If sediments contain low oxygen zones, predators avoid. Trace fossils from the Cambrian onward show increasing burrow complexity, indicating behavioural adaptation alongside anatomical change. Behavioural defences extend beyond burrowing. Some organisms likely adopted nocturnal or low-light activity patterns to reduce detection. Others may have aggregated in groups, gaining safety in numbers. While direct fossil evidence for behaviour is limited, ecological modelling supports these interpretations. Chemical defences probably developed as well. Certain modern marine organisms produce toxins or distasteful compounds. Fossil evidence rarely preserves such chemistry, but evolutionary continuity suggests similar strategies may have existed early on. Shell microstructure analysis provides detailed insight into defensive adaptation. Some shells exhibit layered construction combining strength and fracture resistance. Composite structures resist cracking better than homogeneous materials. This engineering principle appears repeatedly in biological armour. Molting arthropods face particular challenges. During molting, exoskeletons soften temporarily, increasing vulnerability. Some species may have timed molts during safer environmental conditions or hidden during these periods. Fossil assemblages sometimes include clusters of molted exoskeletons, hinting at synchronized molting behavior. The emergence of jaws among early fish intensified defensive pressures further. Jaws allow stronger bites and more precise prey handling. Once jawed predators appeared, prey defences escalated correspondingly. Thick dermal armour in placoderms, for example, reflects both predation and intraspecies competition. Hydrodynamic considerations influence defensive shapes. Smooth shells reduce drag, aiding escape swimming. Conversely, spines increase drag but enhance protection. Trade-offs between mobility and defense shaped morphology. Energy allocation is central to defensive evolution. Producing armour requires resources that could otherwise support reproduction or growth. Species balance these competing demands differently depending on predation intensity and environmental stability. Environmental chemistry influenced mineral availability. Regions with higher calcium concentrations supported more extensive shell formation. Geological evidence correlates mineral abundance with shell diversity patterns. Mass extinction events periodically reset defensive trends. After extinctions, surviving species diversify rapidly, sometimes producing new defensive strategies. This cyclical pattern appears repeatedly throughout the Paleozoic. Reef ecosystems played a special role. Reef structures provide physical shelter, reducing need for heavy armour in some species, while encouraging specialised defences in others. Complex habitats support niche diversification. Predator adaptation did not lag behind. Crushing appendages, stronger jaws, and improved sensory systems evolved in response to prey defences. Fossil evidence shows increasing predator specialisation through the Paleozoic. Some defensive traits had secondary functions. Shells provide not only protection but also structural support for muscle attachment. Spines may assist in stabilisation within currents. Multifunctionality enhances evolutionary viability. Growth patterns recorded in shell layers reveal environmental conditions. Isotope analysis can indicate temperature fluctuations, salinity, and nutrient availability. These environmental factors influence defense investment. Predator-prey dynamics also influenced reproductive strategies. Species under heavy predation often produce more offspring to compensate for losses. Others invest in fewer but better protected young. Geographic variation appears clearly in fossil records. Regions with intense predation pressure show more elaborate defensive morphologies. Comparative studies across basins support this interpretation. Evolution rarely produces perfect solutions. Defensive structures sometimes become so elaborate they hinder mobility or feeding. Such extremes can lead to eventual extinction if environmental conditions shift. Genetic plasticity allows rapid adjustment to predation changes. Populations with greater genetic diversity adapt more readily. Fossil records occasionally show sudden increases in defensive trait frequency, consistent with selective sweeps. Sediment oxygenation from increased burrowing influence microbial ecosystems. These changes cascade through food webs, illustrating how defensive behaviour shapes entire habitats. The interplay between defence and offence also influence biodiversity. High predation pressure often correlates with increased species diversity because specialisation reduces direct competition. Tectonic changes affecting sea level altered habitat availability. Expanding shallow seas encourage diversification, while regression reduces habitat and intensifies competition. Temperature influences metabolic rates, which in turn affect both predator activity and prey defensive capacity. Warmer waters generally increase activity levels. Predator recognition mechanisms likely evolved. Prey capable of detecting predator cues early gain survival advantages. Chemical, visual, and mechanical detection all play roles. Juvenile vulnerability drove some defensive adaptations. Early life stages often lack full armour, so parental investment or habitat selection becomes important. Structural coloration in shells, though rarely preserved, may have contributed to camouflage or signalling. Comparisons with modern marine ecosystems reinforce these interpretations. Similar predator-prey arms races occur today among crustaceans, mollusks, and fish. Ecological modelling shows that introduction of effective predators increases defensive trait prevalence predictably over generations. Functional morphology studies reconstruct how defensive structures performed mechanically. Computer simulations test fracture resistance and hydrodynamics. Biogeochemical cycles respond to increased shell production. Carbon sequestration in shells influences atmospheric carbon dioxide over geological timescales. Extinction selectivity sometimes favours heavily armoured species during certain environmental stresses, though not universally. Predator satiation strategies may have existed. Mass spawning events overwhelm predators temporarily, increasing offspring survival. Fossil assemblages occasionally preserve predator-prey interactions directly, such as bite marks or repaired shell injuries. Repair scars indicate survival after attacks. These scars provide strong evidence of ongoing predation pressure. Defense escalation did not occur uniformly across all taxa. Some groups remained lightly armoured yet survived through behavioural adaptation. Energetic efficiency remains a constant constraint. Excessive armour without sufficient benefit leads to evolutionary disadvantage. Ultimately, defensive evolution reflects ecological balance rather than dominance. Prey species persist not by eliminating predators, but by maintaining survivable interaction levels. These developments transformed Paleozoic oceans from relatively quiet ecosystems into dynamic environments where structural innovation became a key survival factor. As defensive adaptations spread, they created new, selective pressures on predators, encouraging more specialized hunting strategies and further anatomical diversification, and examining one of the most widespread and successful marine groups from that time helps clarify how defense, adaptation, and ecological resilience combine to sustain complex marine ecosystems over immense spans of geological time. If you were able to visit Paleozoic oceans at almost any point during that enormous stretch of time, one group of animals you would repeatedly encounter is trilobites. They were not always the largest animals around, and they were not always top predators, yet their long persistence, diversity, and ecological flexibility make them one of the clearest examples of evolutionary success in early marine history. They appeared early in the Cambrian period and survived for roughly 270 million years before disappearing in the end Permian extinction. That duration alone tells you something important about how well adapted they were. Trilobites belong to the arthropod lineage, meaning they share distant ancestry with modern insects, crustaceans, and arachnids. Their defining feature is the three-lobed body layout that gives them their name. Viewed from above, the body divides into a central axial lobe flanked by two pleural lobes. This arrangement is not merely aesthetic. It reflects underlying segmentation that allowed flexibility, efficient movement along the seafloor, and the ability to curl up defensively. The body divides into three major regions: the cephalon, or head, the thorax made of multiple articulated segments, and the pygidium, or tail section. The number of thoracic segments varied widely among species, and this variation influenced mobility. More segments generally allowed greater flexibility. Fewer segments produce sturdier but less flexible forms. That diversity hints at different ecological roles. One factor behind trilobite success was their exoskeleton. Made primarily of calcite and chitin, it provided both structural support and protection. Calcite is relatively dense, which helps preserve fossils well. This durability is why trilobites dominate many Paleozoic fossil collections. But beyond preservation, the exoskeleton served real biological functions. It protected soft tissues, provided muscle attachment sites, and aided locomotion across sediment. Molting was essential for growth. Like modern arthropods, trilobites periodically shed their exoskeletons. Fossil beds often contain large numbers of discarded shells, sometimes leading early researchers to overestimate population sizes. Molting introduced vulnerability, so trilobites likely sought sheltered areas during these periods. Evidence of healed injuries in some fossils suggests they survived predatory encounters, reinforcing the idea that defensive behavior mattered. Eyes represent another notable adaptation. Many trilobites possessed compound eyes made of calcite lenses. This is unusual because most modern arthropod eyes use organic lenses. Calcite lenses provided rigidity and optical clarity underwater. Some species had large, well-developed eyes suitable for active environments. Others reduced or lost eyes entirely, particularly those living in deeper or murkier waters. That variation demonstrates ecological flexibility. Vision-influenced behavior. Trilobites with large eyes likely relied on visual cues for navigation and predator detection. Reduced eye species may have depended more on tactile or chemical sensing. Sensory diversity supports occupation of varied habitats. Locomotion varied as well. Some trilobites were primarily benthic, meaning they crawled along the seafloor feeding on detritus or small organisms. Others may have swum periodically, using coordinated leg movement and possibly slight body undulations. Limb fossils are rare but indicate jointed appendages capable of walking and feeding simultaneously. Feeding strategies differed among species. Many were deposit feeders, processing sediment to extract organic material. Others appear to have been scavengers. A few may have been active predators of very small organisms. Mouthparts are rarely preserved, but associated trace fossils help infer feeding behaviour. Their enrolment ability remains one of the most effective defensive strategies. By curling into a tight ball, trilobites protected their softer ventral surfaces. Spines on some species enhance this protection. Enrolment also reduces exposed surface area, making it harder for predators to gain purchase. Habitat diversity contributed significantly to their long survival. Trilobites occupied shallow coastal waters, reef environments, continental shelves, and deeper offshore zones. Some specialized in particular sediment types. Others tolerated broader environmental ranges. Generalists often survive environmental fluctuations better than specialists. Reproductive strategies remain largely speculative because direct evidence is scarce. However, many arthropods release large numbers of eggs. High reproductive output increases resilience against predation and environmental stress. Trilobite diversity peaked during the Ordovician period. Hundreds of genera existed simultaneously, each adapted to slightly different ecological niches. This diversification suggests stable ecosystems capable of supporting complex food webs. Environmental changes periodically reduce trilobite diversity. Climate shifts, sea level changes, and mass extinction events all took tolls. Yet trilobites repeatedly rebounded, demonstrating adaptability. Competition likely influenced their evolution. Other arthropods, early fish, and various invertebrates competed for resources. Competitive pressure encourages specialization. Sediment interaction offers another interesting angle. Trilobite movement and feeding disturbed sediment layers. This bioturbation influenced oxygen penetration and microbial distribution. Thus, trilobites shaped habitats as well as inhabited them. Shell ornamentation varied widely. Smooth shells reduced drag in active species. Ribbed or spiny shells offered protection or stabilization in currents. These structural differences reflect environmental adaptation. Some trilobites show evidence of sexual dimorphism, meaning males and females differed slightly in size or morphology. Such differences can reflect reproductive strategies. Fossil trackways provide behavioral evidence. Distinctive crawling traces attributed to trilobites reveal movement patterns, feeding routes, and group behavior. Group behavior itself is suggested by mass fossil assemblages. Some deposits contain numerous individuals of similar size. Possible explanations include molting gatherings, spawning events, or environmental trapping. Trilobite extinction patterns illustrate ecological vulnerability. While they survived multiple earlier extinction events, the end Permian event proved catastrophic. Rapid environmental changes, including ocean anoxia and temperature shifts, exceeded their adaptive capacity. Comparisons with modern arthropods help interpret trilobite biology. Horseshoe crabs, for example, share some structural similarities and long evolutionary persistence, though they are not direct descendants. Calcite eye lenses present interesting optical physics. Their refractive properties suit underwater environments well. Some trilobite eyes even show correction for spherical aberration, indicating sophisticated optical evolution. Trilobite larvae likely differed significantly from adults. Ontogenetic studies show developmental stages preserved in fossil series. Changes during growth reflect ecological role shifts. Predator interactions remain a major theme. Bite marks, repaired injuries, and defensive morphology all indicate constant predation pressure. Geographic distribution was global. Trilobite fossils occur on every continent, reflecting widespread marine habitats. Paleogeographic reconstructions show trilobites responding to continental drift. Species distribution shifted as ocean basins reconfigured. Isotopic analysis of shells provides clues about ancient water temperatures and chemistry, indirectly informing trilobite ecology. Evolutionary turnover within trilobites was continuous. New genera replaced older ones gradually. This turnover reflects adaptation to changing conditions. Taxonomic diversity sometimes masks ecological continuity. different species may perform similar ecological roles despite morphological differences. The durability of trilobite exoskeletons contributes to their prominence in museums and educational materials, shaping public perception of Paleozoic life. Despite their success, trilobites were never alone. They shared ecosystems with brachiopods, mollusks, echinoderms, early fish, and numerous soft-bodied organisms. Energy flow through ecosystems often placed trilobites at intermediate trophic levels. Not always predators, not always prey exclusively. Their adaptability illustrates a broader principle. Evolutionary success depends on flexibility, reproductive resilience, and ability to occupy multiple niches. Long survival does not guarantee indefinite persistence. Environmental thresholds eventually surpass adaptation capacity. Understanding trilobites helps contextualize early marine ecosystems. They connect predator emergence, defensive evolution, sediment dynamics, and biodiversity expansion. Their story also reminds us that dominance in evolutionary terms means persistence rather than size or aggression alone. As marine ecosystems continued evolving, other sensory adaptations began shaping survival strategies, particularly visual systems that dramatically altered predator-prey interactions, and looking at how vision advanced in those ancient seas helps clarify another major step in the increasing complexity of Paleozoic life. For a long time in Earth's history, light existed without being interpreted in any complex way. Sunlight filtered through shallow water, scattered across sediment, reflected off suspended particles, and faded into darkness with depth. Organisms responded to light chemically and biologically, adjusting metabolism or orientation, but they did not truly see. The development of vision in the Paleozoic ocean marked one of the most transformative changes in ecological interaction. Once animals could detect shapes, movement, contrast, and direction, the rules governing survival shifted dramatically. Light behaves differently underwater than in air. Water absorbs longer wavelengths quickly, so red disappears first, followed by orange and yellow. Blue penetrates deepest. In shallow Paleozoic seas, where much early diversification occurred, light conditions were suitable for visual detection at moderate distances. That environmental reality provided opportunity for selection to favour organisms capable of forming images rather than simply sensing brightness. The earliest visual systems were likely simple photoreceptive patches. These clusters of light-sensitive cells allowed organisms to distinguish light from dark and perhaps detect shadow movement. Even that limited capability provides survival advantage. Detecting an approaching shadow could signal predator presence. Detecting bright areas could guide movement toward productive zones. Gradually, structural refinements improve directionality. Shallow depressions around photoreceptive cells reduce light from unwanted angles, increasing spatial resolution. With further development, these depressions deepen, eventually forming pinhole-like structures capable of forming crude images. Lens development improves focus, increasing clarity. The Cambrian fossil record contains clear evidence of advanced eyes in certain arthropods. Trilobites in particular display compound eyes composed of numerous individual lenses. Each lens captures a portion of the visual field. Combined, they create a mosaic image suitable for detecting motion and contrast. Motion detection is especially important underwater, where turbidity often limits clarity. Calcite lenses in trilobite eyes represent an unusual evolutionary solution. Most modern arthropods use organic materials for lenses. Calcite provides rigidity and transparency underwater, and its crystalline structure can focus light effectively when properly shaped. Some trilobite lenses show correction for optical distortions, indicating refinement beyond simple light detection. The presence of complex eyes implies neural processing capacity. Visual information must be integrated, interpreted, and translated into movement. That requires centralization of nervous systems. Evidence from fossil morphology suggests increasing cephalization, meaning concentration of sensory organs and neural tissue at the head region. Cephalization correlates strongly with active locomotion and predation. Predators benefit greatly from vision. Detecting prey at distance allows planning approach trajectories. Visual tracking supports coordinated grasping movements. Once predators evolve effective sight, prey species face new selective pressures. Camouflage, rapid escape responses, burrowing behavior, and defensive armor become more valuable. Prey species also benefit from vision. Early detection of predators improves survival odds. Visual scanning of environment allows avoidance strategies. Thus, both sides of predator-prey interactions escalate sensory capabilities. Vision also influences habitat preference. Clear shallow waters favour image-forming eyes. Murkier or deeper environments may favour reduced eyes or alternative sensory systems, such as mechanoreception or chemoreception. Fossil records show trilobite species with large eyes dominating certain shallow environments, while reduced eye species occupy deeper or sediment-rich zones. The emergence of visual predators altered behavioural patterns. Instead of random movement, animals began responding to visible cues. Directional pursuit replaced opportunistic feeding. Group behaviours may have developed as well, since vision allows recognition of conspecifics at distance. Coloration patterns, though rarely preserved in fossils, likely evolved in response to vision. Camouflage patterns help blend with substrate. Counter-shading, darker dorsal surfaces and lighter ventral surfaces, reduces detectability. Modern marine organisms demonstrate these strategies widely, suggesting early origins. Environmental light gradient structured vertical distribution of species. Some organisms remained in well-lit zones, others descended into dimmer waters to avoid detection. Vertical migration patterns may have emerged early in response to visual predation. Visual competition also occurred among predators. Those with sharper motion detection or wider fields of view gained advantage. Compound eyes naturally provide wide fields, reducing blind spots. Water clarity in Paleozoic seas varied regionally. Areas with high plankton density reduce visibility, affecting selective pressure on eye development. Fossil evidence suggests variability in eye size and complexity correlates with environmental conditions. Neural complexity associated with vision likely influenced other cognitive capabilities. Coordinated movement, obstacle avoidance, and environmental mapping become more refined with visual input. Some Cambrian predators, such as Anomalocaris, possessed large compound eyes relative to body size. This indicates heavy reliance on sight. Eye placement suggests broad forward and lateral coverage. The presence of image-forming eyes also influences diel cycles. Visual hunters often rely on daylight. Nocturnal niches open for species adapted to low-light conditions. Mechanoreception remains important. Even visually oriented species use vibration detection. However, vision allows prediction rather than mere reaction. Evolution of eyes likely occurred multiple times independently across different lineages. Convergent evolution demonstrates the strong advantage of sight in complex ecosystems. Light-driven behavior influences reproductive strategies. Some species may time spawning to certain light conditions to reduce predation risk. Hydrodynamic wake detection combined with visual tracking enhances hunting efficiency. Integration of sensory modalities increases success. Developmental genetics underlying eye formation are highly conserved across modern animals. This suggests deep evolutionary roots extending to early complex organisms. Predator-prey arms races intensified as vision improved. Faster prey favoured streamlined bodies. Heavier armour favoured crushing appendages in predators. Increased mobility associated with visual tracking reshaped sediment dynamics. Active swimming and rapid escape disturb substrates more frequently. Behavioural fossils, such as escape traces or directional burrows, may reflect visually mediated interactions. visual acuity limitations likely constrained maximum hunting distance. Water absorbs light, so even advanced eyes operate within restricted range. Deep-sea environments remained largely visually limited. Species there continued relying on alternative senses. Light polarization detection may have evolved early, aiding orientation in water. Shadow casting becomes ecologically meaningful once organisms interpret it. Predators may approach from angles minimizing detection. Bioluminescence appears later in evolutionary history, but eventually interacts with visual systems extensively. Energy cost of maintaining complex eyes influences trade-offs. Species in low-light habitats often reduce eye size to conserve energy. Predation intensity correlates with eye development diversity in fossil assemblages. Eye loss in some lineages suggests habitat shifts rather than regression in complexity. Cephalization continues intensifying beyond the Cambrian, laying groundwork for later vertebrate sensory systems. Vision contributed to emergence of more sophisticated ecosystems with layered trophic interactions. As Paleozoic oceans progressed into later periods, visual systems continued refining. Jawed fish developed camera-type eyes distinct from arthropod compound eyes. These parallel visual strategies demonstrate that image formation is a powerful evolutionary driver across unrelated groups. Once sight becomes central to survival, ecosystem structure transforms permanently. Behavioral strategies, morphology and habitat use all reorganize. Understanding how vision emerged clarifies why later Paleozoic seas supported increasingly active predators, and as these visual systems integrated with new anatomical innovations like articulated limbs and improved swimming structures, marine ecosystems advanced toward even greater complexity that would define the next phases of ocean evolution. As marine ecosystems continued becoming more structured and competitive, some arthropods began moving into roles that demanded larger bodies, stronger appendages, and increasingly specialized sensory equipment. Arthropods already had a successful basic design: segmented bodies, jointed limbs, external skeletons, and flexible developmental pathways that allowed variation without compromising overall function. That flexibility made them especially capable of adapting when ecological opportunities appeared. During later Paleozoic periods, particularly the Silurian and Devonian, this adaptability produced some of the most imposing marine arthropods known, including the group commonly called sea scorpions or eurypterids. Eurypterids were not true scorpions, although they share distant arthropod ancestry. The nickname comes mostly from general appearance: elongated bodies, prominent pincers in some species, and occasionally a tail spine. Their anatomy actually reflects a mix of adaptations for swimming, crawling along sea floors, and in some cases possibly brief excursions into very shallow or brackish environments. Fossil evidence suggests considerable diversity within this group, with species ranging from relatively small forms under 20 centimeters long to giants exceeding 2 meters. Size increase in arthropods often depends on environmental oxygen levels. During parts of the Paleozoic, atmospheric oxygen concentrations rose significantly compared with the earlier periods. Arthropods rely partly on diffusion-based respiratory systems, so higher oxygen availability can support larger body size. Marine environments also provide buoyancy, reducing mechanical stress on large exoskeletons. Together, these factors allowed certain arthropods to reach dimensions rarely seen in their modern marine relatives. The exoskeleton remained central to their success. Composed of chitin reinforced with minerals, it provided both protection and leverage for muscle attachment. Larger eurypterids show particularly thickened cuticle regions near limb bases, suggesting strong musculature for grasping prey or maneuvering in water. Molting remained necessary for growth, which introduced periodic vulnerability, but the advantages of structural support outweighed the risks. Swimming adaptations varied. Some eurypterids possessed paddle-like appendages on posterior limbs. These flattened structures resemble modern swimming legs in crustaceans. Hydrodynamic modelling suggests these paddles could generate efficient thrust, enabling active pursuit of prey or escape from larger predators. Other species retained limbs better suited for walking along sediment, indicating ecological partitioning within the group. The head region typically featured compound eyes, often positioned laterally for broad fields of view. Visual capability, already important in earlier Paleozoic ecosystems, remained crucial. Detecting motion across open water or along the seafloor aids both hunting and predator avoidance. Eye size varies among species, likely reflecting habitat clarity and depth preferences. Feeding structures also diversified. Some eurypterids possessed large grasping chelicerae, capable of capturing sizable prey relative to their own body length. Others had more delicate appendages suggesting scavenging or feeding on smaller organisms. Fossilized gut contents are rare, but provide occasional confirmation of diet. Predatory impact on ecosystems becomes clearer when examining prey morphology during the same intervals. Increased shell thickness, spine development, and enrollment behavior among trilobites coincide with eurypterid diversification. This parallel evolution indicates sustained predation pressure. Seafloor interaction patterns changed as well. Large arthropods crawling across sediment disturb substrates significantly. This disturbance influences microbial communities, oxygen penetration, and nutrient redistribution. Bioturbation thus increases not just from burrowing species, but also from mobile predators. Some eurypterids inhabited brackish or freshwater environments. Fossils from ancient river systems and lagoons support this interpretation. Movement between marine and marginal habitats demonstrates ecological flexibility. Such transitions may have provided refuges during marine environmental stress. Reproductive behavior remains largely speculative, but comparisons with modern arthropods suggest possible egg-laying in protected environments. Shallow waters or sheltered sediment zones would offer some protection from predation. Coloration is unknown due to fossilization limits, yet camouflage likely played a role. Sediment-matching coloration helps ambush predators remain undetected. Hydrodynamic body shape differs among species. Streamlined forms suggest active swimmers. Broader, flatter bodies indicate benthic lifestyles. Morphological diversity reflects ecological specialization. Competition among predators likely intensified during this period. Early jawed fish emerged alongside eurypterids. Different predatory strategies reduced direct competition. Fish relied on jaws and fins. Arthropods relied on appendages and exoskeleton leverage. Climate conditions influenced distribution. Warmer shallow seas supported greater biodiversity. Cooling events sometimes contracted suitable habitats. Salinity tolerance appears broad in certain eurypterids. Adaptability to fluctuating salinity enhances survival in coastal environments. Oxygen stratification in water columns may have limited vertical distribution. Larger arthropods generally occupied well-oxygenated zones. Fossil trackways attributed to eurypterids show walking patterns, resting marks, and occasional swimming traces preserved in sediment. These behavioural fossils provide insight into locomotion and habitat use. Growth series preserved in some fossil beds allow reconstruction of developmental stages. Juvenile morphology sometimes differs significantly from adults, suggesting niche shifts during growth. Predation risk for juvenile eurypterids likely remained high. Smaller size increases vulnerability even for predatory species. Exoskeleton ornamentation includes spines, ridges, and sculptured surfaces. Some features may enhance structural strength. Others could deter predators or competitors. Energy requirements for large arthropods necessitate productive ecosystems. High plankton productivity supports food chains leading to apex predators. Taphonomic bias again influences perception. Hard exoskeletons fossilize readily. Soft-body contemporaries remain underrepresented. The evolutionary peak of eurypterids occurred during the Silurian and Devonian periods. Afterward, diversity declined gradually. Changing climates, competition with vertebrate predators and habitat shifts likely contributed. Mass extinction events reduced many arthropod lineages. Eurypterids survived several early crises but did not persist beyond the Permian. Despite extinction, their ecological role influenced later marine evolution. Predatory niches established by arthropods were later occupied by vertebrates. Biomechanical analysis suggests strong grasping force in certain species. Appendage articulation allowed effective prey manipulation. Water temperature affects metabolic rates. Warmer conditions generally increase activity levels in ectothermic organisms like arthropods. Nutrient cycling accelerates with increased predation. More biomass moves through food webs. Evolutionary arms races remain evident. Prey defenses intensify alongside predator specialization. Ecosystem stability often correlates with predator presence. Apex predators regulate intermediate populations. Sedimentary evidence shows increasing complexity in community structure during Eurypterid dominance. Continental drift repositioned habitats. Distribution patterns shifted accordingly. Isotope studies of associated sediments provide environmental context, including salinity and temperature estimates. Comparisons with modern horseshoe crabs offer limited analogies, mainly regarding exoskeleton function and molting behavior. Despite their size, Eurypterids were not invincible. Fossils show injuries, possible predation marks, and molting-related mortality. Ecological diversity among arthropods prevented monoculture dominance. Multiple predatory species coexisted. The broader significance lies in how arthropods demonstrated the potential for large-bodied predation before vertebrates dominated oceans. Understanding these giants clarifies that Paleozoic marine ecosystems supported multiple evolutionary pathways toward large predator roles. As marine biodiversity continued expanding through the Ordovician and into later periods, ecological interactions became even more layered, and examining how entire ocean communities diversified during that time helps connect individual predator stories to broader environmental patterns shaping early complex life. By the time the Ordovician period began, roughly 485 million years ago, marine ecosystems had already undergone significant change compared with the quieter, earlier Paleozoic oceans. The Cambrian diversification had introduced complex body plans, active predators, improved sensory systems, and increasingly structured food webs. What followed in the Ordovician was not a sudden burst like before, but a broad expansion. Marine biodiversity increased steadily, ecosystems filled out with new species, and interactions became more layered. The process was less dramatic on the surface than the Cambrian rise in complexity, yet it proved just as important because it stabilized and refined marine ecological systems. Sea levels during much of the Ordovician remained high. Large portions of continental crust sat underwater, beneath shallow epicontinental seas. These shallow seas created ideal conditions for biodiversity. Sunlight reached the seabed, nutrient input from weathering continents remained steady, and water circulation distributed oxygen effectively. Such environments tend to produce high biological productivity even today, and the Ordovician was no exception. Temperature patterns also supported diversification. Many regions experienced relatively warm conditions. Warmer water increases metabolic rates for ectothermic marine organisms, encouraging activity, growth, and reproduction. At the same time, stable temperature ranges reduce environmental stress, allowing ecosystems to mature over long periods. One noticeable development during the Ordovician was the expansion of reef-like communities. Earlier reef structures existed, but they became more widespread and complex during this period. Organisms such as stromatoporoids, bryozoans, corals, and calcareous algae contributed to building rigid frameworks. These reef environments created microhabitats, sheltered zones, and concentrated food sources. Habitat complexity generally correlates with species diversity because different organisms exploit different niches within the same structure. brachiopods flourished particularly well. These shelled marine animals superficially resemble clams but belong to a distinct lineage. Their diversity increased dramatically during the Ordovician. Many were filter feeders, extracting suspended organic particles from water. Their success reflects abundant plankton and stable water chemistry. Trilobites remained common but began sharing dominance with other groups. Their relative ecological prominence shifted slightly as new organisms diversified. This illustrates a broader trend: ecosystems transitioning from dominance by a few pioneering groups to more balanced multi-group communities. Mollusks diversified extensively. Early cephalopods, ancestors of modern squids and octopuses, appeared in more advanced forms. Some developed straight shells, others slightly curved. These shells provided buoyancy control, enabling vertical movement through water columns. Active predation from cephalopods added another dimension to marine food webs. Echinoderms also diversified. Starfish-like organisms, crinoids, and other relatives occupied various ecological roles. Many anchored themselves to substrates, filter feeding with specialized appendages. Others moved more freely across sediments. Plankton communities expanded as well. Increased plankton diversity enhances energy transfer through ecosystems. Higher productivity at the base supports more complex, higher trophic levels. Quiet competition characterized much of this expansion. Instead of dramatic predator-prey escalation alone, species competed for space, nutrients, and stable environmental niches. Competition often drives specialization. Slight differences in feeding strategy, habitat preference, or reproductive timing reduce direct conflict and allow coexistence. Sediment interactions became increasingly complex. Burrowing organisms mix sediments more thoroughly. This process improved oxygen penetration and altered microbial communities. Enhanced sediment oxygenation supports more diverse benthic life. Water chemistry remained conducive to shell formation. Calcium carbonate saturation allowed continued biomineralization. Shell thickness and ornamentation increased in several groups. Ocean circulation patterns stabilized in many regions due to continental configurations. Stable currents support consistent nutrient distribution. Consistency favors long-term ecosystem development. Predation still existed, but often in a balanced state. Apex predators were present, yet prey species maintained viable populations through defensive adaptations, reproductive output, and habitat use. Visual systems continued evolving. Improved eyesight among predators and prey refined behavioral interactions. Camouflage strategies likely intensified, though direct fossil evidence remains limited. Sound detection and vibration sensing probably played increasing roles. Water transmits mechanical signals efficiently, and organisms capable of detecting subtle disturbances gain survival advantages. Reproductive diversification accelerated. Some species released free-swimming larvae. Others attached eggs to substrates. Diverse strategies increase resilience against environmental variability. Geographic provinciality emerged clearly. Different marine basins developed distinct faunas. Continental separation limited species mixing. Regional specialization followed. Plate tectonics influenced habitat connectivity. Opening and closing ocean basins periodically altered migration routes. Carbon cycling intensified as shell-producing organisms proliferated. Burial of carbonate shells influences long-term climate regulation. Oxygen levels in shallow seas generally remained sufficient for active organisms. Deeper basins sometimes experience low oxygen conditions, restricting large animal habitation there. Climate fluctuations toward the late Ordovician introduced stress. Evidence suggests cooling trends and glaciation events near the period's end. These environmental changes triggered one of the first major mass extinction events affecting marine life. Before that extinction, however, ecosystems had reached remarkable diversity. Fossil assemblages show multiple trophic levels, varied body plans, and widespread ecological interdependence. nutrient availability remained a central driver. Weathering of continental rocks released phosphorus and other nutrients into oceans. Nutrient-rich waters sustain plankton, supporting entire food webs. Salinity variations occurred in restricted basins. Some organisms adapted to fluctuating salinity, expanding ecological tolerance. Storm disturbances periodically reshaped shallow habitats. While destructive locally, storms also create new colonization opportunities. Energy efficiency and feeding strategies improved. Filter feeding apparatus became more specialized. Some organisms optimized particle capture through complex appendage structures. Biological interactions extended beyond predation and competition. Symbiosis likely became more common. Certain organisms benefited from close association with others, such as shelter provision or nutrient exchange. Metabolic diversity expanded. Some organisms tolerated low oxygen. Others required well-oxygenated water. This variation structured vertical habitat use. Population densities increased in productive areas. High density encourages both competition and evolutionary innovation. Trace fossils from Ordovician sediments show diverse movement patterns. Complex burrows, resting traces, and feeding marks indicate varied behaviours. The ecological stability achieved during much of the Ordovician allowed gradual refinement rather than abrupt change. That refinement built resilient ecosystems capable of supporting large-bodied organisms. The eventual late Ordovician extinction reminds us that stability is never permanent. Environmental thresholds, once crossed, can reset ecosystems dramatically. Yet even extinction events contribute to long-term diversification. Survivors occupy vacant niches, often producing new evolutionary pathways. From a broader perspective, Ordovician seas demonstrate how complexity matures after initial diversification. Instead of explosive innovation alone, steady ecological structuring produces sustainable biodiversity. Understanding this period helps clarify how Paleozoic oceans transitioned from experimental early ecosystems to established marine communities. These established communities set the stage for further evolutionary developments, including increasingly sophisticated reef systems, continued predator specialization, and eventual emergence of jawed vertebrate dominance, all of which become easier to understand when viewed against the backdrop of expanding, quietly competitive Ordovician marine ecosystems that were already shaping the structure of life beneath the surface. Long before fish became the dominant visual symbols of marine life, reef ecosystems were already developing beneath Paleozoic seas. These early reef communities did not resemble modern coral reefs exactly, yet they performed similar ecological functions. They created structure where open seafloor had previously dominated, offered shelter to smaller organisms, concentrated food resources, and provided stable attachment points for species that depended on fixed positions in moving water. In practical terms, these reefs acted like underwater cities, not in a metaphorical sense, but as dense, layered habitats, where many species lived in close proximity and interacted constantly. The earliest substantial reef frameworks appeared during the Ordovician period and expanded significantly into the Silurian and Devonian. Unlike modern reefs built primarily by scleractinian corals, early Paleozoic reefs often relied heavily on stromatoporoids, tabulate corals, rugose corals, bryozoans, calcareous algae, and microbial communities. Each contributed structural material in slightly different ways. Stromatoporoids, for example, were sponge-like organisms that secreted layered calcium carbonate skeletons. Over time, these skeletons accumulated, forming rigid frameworks capable of resisting wave action. Calcareous algae played an equally important role. By depositing calcium carbonate as they grew, these algae helped cement reef structures together. Microbial mats stabilized sediment surfaces, reducing erosion. The interaction of biological construction and sediment stabilization allowed reefs to grow vertically and horizontally. Water clarity strongly influenced reef formation. Photosynthetic organisms require light, so reefs generally formed in shallow sunlit environments. Epicontinental seas covering continental interiors during the Paleozoic provided ideal conditions: warm temperatures, moderate salinity, and consistent nutrient supply supported sustained growth. Reefs create microhabitats through physical complexity. Crevices, overhangs, sheltered cavities, and elevated surfaces all offer different environmental conditions. Some species prefer calm water pockets, others thrive where currents deliver food continuously. This structural diversity drives biodiversity. Filter feeders dominated many early reef communities. Organisms such as brachiopods, bryozoans, and certain sponges extracted plankton and organic particles from passing water. Their feeding activity in turn influenced local water chemistry and nutrient distribution. Predation within reef systems existed but often differed from open water hunting. Ambush strategies became common. Organisms hiding within reef cavities waited for prey to approach. Defensive armour among reef inhabitants remained important due to close quarters. Sediment trapping is another key reef function. Reef frameworks slow water movement locally, causing suspended particles to settle. Sediment accumulation supports additional benthic organisms. Over time, entire carbonate platforms develop from this process. Chemical cycling intensifies in reef environments. Photosynthesis by algae increases oxygen locally during daylight. Respiration by animals consumes oxygen at night. These daily cycles influence community composition. Temperature stability often improves within reef structures. Complex frameworks buffer sudden environmental changes, providing refuges during storms or temperature fluctuations. Competition for space becomes intense in reef environments. Organisms grow upward or outward to secure light and water flow. Overgrowth interactions leave identifiable patterns in fossilized reef surfaces. Some reef organisms developed specialized attachment structures. Holdfasts, cementing secretions, and encrusting growth forms all help maintain position against currents. Bioerosion also occurred. Certain organisms bored into reef skeletons, creating cavities that later served as shelter for other species. This process contributes to habitat complexity. Symbiotic relationships likely existed even in early reefs. Although direct fossil evidence is limited, modern analogues suggest nutrient exchange between different organisms provided mutual benefits. Hydrodynamics influence reef morphology. Wave-exposed reefs develop robust frameworks, while sheltered reefs show more delicate structures. Reef expansion contributes significantly to carbonate deposition in geological records. Large limestone formations often trace back to ancient reef systems. Predator diversity in reef habitats increased gradually. Arthropods, early cephalopods, and eventually fish exploited concentrated prey populations. Mobility patterns varied widely. Some organisms remained permanently attached. Others moved slowly across reef surfaces. A few actively swam above reef structures. Larval dispersal played a central role in reef colonization. Many marine organisms release planktonic larvae capable of drifting before settling. This process connects reef communities across regions. Salinity stability is important for reef health. Extreme fluctuations limit growth. Paleozoic epicontinental seas often maintained relatively stable salinity. Oxygen availability tends to remain high in shallow reef waters due to wave mixing. This supports active organisms. Nutrient balance must remain moderate. Excess nutrients encourage algal overgrowth that can smother reef builders. Evidence suggests many Paleozoic reefs existed under balanced nutrient conditions. Storm damage periodically reshaped reefs. Broken skeletal fragments often became building blocks for new growth. Fossil reef cross-sections reveal successive growth layers, showing long-term ecological continuity. Climate changes influenced reef distribution. Cooling events reduced tropical shallow seas, contracting reef habitats. Mass extinctions affected reef builders significantly. Some reef-forming groups disappeared entirely, replaced later by different organisms. Reef ecosystems illustrate early complex ecological networking. Producers, consumers, decomposers, and structural engineers all interacted closely. The absence of dominant fish during early reef development meant arthropods, mollusks, and other invertebrates filled many ecological roles now associated with vertebrates. Energy flow in reef systems tends to be efficient. Close proximity of organisms reduces energy loss between trophic levels. Behavioral specialization increased in reef habitats. Camouflage, burrowing, attachment, and territorial behaviors likely emerged. Chemical signaling becomes important in dense communities. Reproductive timing, territorial defense, and predator warnings may involve chemical cues. Water flow patterns around reefs influence feeding efficiency. Some organisms orient themselves strategically to maximize particle capture. Reef frameworks influence local sedimentation patterns, shaping surrounding habitats. Long-term reef persistence contributes to geological stability in certain marine regions. Comparisons with modern reefs highlight continuity in ecological principles, despite differences in dominant organisms. The term strange cities reflects density and complexity rather than human-like organization. Multiple species coexist closely with constant interaction. Reefs provided evolutionary testing grounds. Close contact accelerates competition and innovation. Evidence of disease or parasitism in fossil reefs suggests biological pressures beyond predation. Mineral composition of reef skeletons reflects seawater chemistry at the time of formation. Geochemical signatures from reef limestones help reconstruct ancient ocean conditions. As fish eventually diversified, reef ecosystems changed again. Vertebrate predation introduced new pressures. Yet the fundamental role of reefs as biodiversity hubs remained consistent. Understanding early reef systems clarifies how marine ecosystems developed layered complexity before vertebrate dominance. That layered complexity created environments where new predator types, particularly early jawed fish, could evolve successfully, and examining how those vertebrates entered already structured ecosystems helps explain the next stage in Paleozoic ocean development. As marine ecosystems continued becoming more structured and crowded, another major anatomical innovation began changing predator-prey dynamics in a lasting way. Vertebrates had already appeared earlier in simpler forms, mostly jawless fish that filtered particles from water or fed on small organic material. These early fish were important ecologically but not usually dominant predators. The development of jaws, however, altered that balance. Once vertebrates acquired the ability to bite, grip, and process larger prey efficiently, marine food webs reorganized again. The origin of jaws is generally linked to modifications of anterior gill arches. Structures originally supporting respiration gradually evolved into hinge supports capable of movement. Musculature and connective tissues adapted to generate force. This transformation did not happen abruptly. Transitional fossils show intermediate stages where skeletal supports strengthened before true biting capability emerged. Jawed fish, often called gnathostoms, first diversified noticeably during the Silurian period and expanded dramatically through the Devonian. Their arrival introduced a different predatory strategy compared with arthropods like eurypterids. Arthropods relied on external appendages and exoskeleton leverage. Fish combined internal skeletons, powerful muscles, streamlined bodies, and jaws capable of rapid closure. That combination allowed more sustained swimming and flexible feeding. Internal skeletons made of cartilage or early bone offered advantages. Unlike rigid exoskeletons that must be molted for growth, internal skeletons grow continuously. This allows smoother growth patterns and potentially larger body size over time. Cartilaginous skeletons also reduce weight, improving swimming efficiency. Body shape in early jawed fish often shows streamlining suited for active movement through water. A fusiform body reduces drag. Fins provide stability, steering, and propulsion. These features enhance pursuit capability, allowing predators to chase prey rather than rely primarily on ambush. Sensory systems improved in parallel. Lateral line systems, capable of detecting water movement, help fish sense nearby organisms even in low visibility. Combined with vision, this creates a multi-sensory hunting approach. Teeth represent another key development. Early jawed fish possessed simple tooth-like structures initially, but diversification followed quickly. Some species evolved sharp, conical teeth suited for grasping slippery prey. Others developed crushing plates for armored organisms. Dental specialization reflects dietary diversity. Predation pressure from jawed fish influenced existing marine organisms significantly. Trilobites, brachiopods, mollusks, and other invertebrates show increased shell thickness and defensive ornamentation during periods of fish diversification. This correlation supports the idea of an escalating evolutionary arms race. Placoderms, one of the earliest major jawed fish groups, illustrate this shift well. Many placoderms possessed heavy dermal armor covering head and thoracic regions. Some species, such as large arthropods, reached considerable sizes relative to earlier vertebrates. Their jaw structures could generate substantial force, making them effective predators. Despite heavy armor, placoderms were often active swimmers. Jointed armor plates allowed head movement relative to the body, improving feeding flexibility. Fossilized bite marks on contemporaneous organisms suggest active predation. Cartilaginous fish ancestors also appeared during this time. Though early representatives differ from modern sharks, they already displayed features associated with active predation: streamlined bodies, multiple fins, and sensory adaptations suited to open water hunting. Competition between arthropod predators and vertebrate predators likely influenced evolutionary trajectories on both sides. Vertebrates, with internal skeletons, could achieve different locomotion patterns than arthropods. Arthropods retained advantages in exoskeleton protection and appendage manipulation. Prey organisms responded through multiple strategies. Increased burrowing behavior provided refuge. Shell ornamentation intensified. Some species adopted faster reproductive cycles to offset predation losses. Environmental factors supported vertebrate diversification. Rising oxygen levels during certain Paleozoic intervals allowed higher metabolic activity. Active swimming requires efficient oxygen transport systems. Vertebrate circulatory adaptations facilitated this. Water temperature also affects metabolism. Warmer waters promote faster physiological processes. Devonian seas, often described as warm and productive, provided favourable conditions. Nutrient availability remained high in many shallow marine environments. Plankton productivity supported complex food chains necessary for sustaining large predators. Reef ecosystems provided important habitats for early jawed fish. Structural complexity offers hunting grounds, shelter, and breeding sites. Fish could exploit niches within reef frameworks. Geographic diversification occurred as continental configuration shifted. Isolated basins encourage speciation. Fossil records show regional differences in fish assemblages. Reproductive strategies among early jawed fish remain incompletely understood, but internal fertilization appears to have evolved in some lineages. This adaptation increases offspring survival in certain environments. Hydrodynamic efficiency improved as fin placement and body proportions diversified. Tail fin shapes reflect swimming styles, from cruising to rapid bursts. Neural complexity increased alongside sensory development. Coordinated swimming, schooling behavior, and predation require advanced neural processing. Schooling itself may have evolved partly as predator avoidance. Groups reduce individual predation risk while improving environmental awareness. Jaw mechanics diversified widely. Some fish developed protrusible jaws extending forward to capture prey. Others specialized in crushing hard shells. Stable isotope analysis of fossil fish remains provides dietary clues. Carbon and nitrogen ratios indicate trophic levels. Trace fossils, including feeding marks and coprolites, contribute additional evidence of vertebrate predation. Ecological layering intensified as vertebrate predators occupied mid to upper trophic levels. This layering supports ecosystem resilience but also complexity. Mass extinction events periodically reduced vertebrate diversity, yet jawed fish repeatedly diversified after environmental recovery. Comparisons with modern marine ecosystems show continuity in predator-prey dynamics. Many principles established during the Paleozoic persist today. Vertebrate mobility allowed expansion into open water pelagic zones, beyond benthic habitats dominated earlier. Biogeochemical cycles responded to increased vertebrate biomass. Nutrient recycling accelerated through predation and waste production. Energy transfer efficiency improves with active predators controlling intermediate consumers. The balance between vertebrate and invertebrate dominance shifted gradually. Neither group eliminated the other, but ecological roles redistributed. Evolutionary innovation in jaws paved the way for later terrestrial vertebrate evolution. Feeding versatility supports habitat transitions. Predator diversity contributes to ecosystem stability by preventing overpopulation of specific prey species. Environmental variability, including sea level fluctuations and climate shifts, continued shaping evolutionary outcomes. Understanding the arrival of jawed fish clarifies how marine ecosystems transitioned toward vertebrate prominence without eliminating earlier successful groups. This phase of ecological escalation set the stage for even larger armoured fish that would dominate certain Devonian seas, and examining those Placoderm giants more closely helps illustrate how defensive armour and predatory capability combined in one lineage during a particularly dynamic period of marine evolution. By the time the Devonian period was underway, marine ecosystems had already experienced multiple waves of innovation. Arthropod predators had risen and diversified. Jawed fish had appeared and begun reshaping food webs. Reef systems had matured and spread. What distinguishes the Devonian, however, is the scale and intensity of vertebrate dominance in certain environments. Among the most striking representatives of that shift were the Placoderms, a diverse group of armoured jawed fish that occupied a wide range of ecological roles and in some cases reached impressive sizes. Placoderms first appeared in the Silurian, but they reached their peak diversity and ecological prominence during the Devonian. Their defining feature was heavy dermal armour covering the head and thoracic region. Unlike the flexible scales seen in later fish, Placoderm armour consisted of large bony plates joined by articulations. This plating protected vital organs and supported strong jaw musculature. The armour did not extend across the entire body in most species, allowing flexibility and efficient swimming in the posterior region. One of the most well-known placoderms is Dunkleosteus, an arthrodire placoderm estimated to reach lengths of 6 to 8 metres in some individuals. While size estimates vary, even conservative figures place it among the largest predators of its time. Its head armour formed a rigid structure capable of withstanding significant mechanical stress. The jaw apparatus, rather than bearing true teeth in the modern sense, possessed sharpened bony plates that functioned like blades. Biomechanical analysis suggests these jaws could generate powerful bite forces relative to body size. The articulation between head and thoracic armour in arthrodires allowed vertical movement of the head shield during jaw opening. This mechanical arrangement increased gape size and improved feeding efficiency. Combined with strong musculature, this system enabled placoderms to process sizable prey. Not all placoderms were giant apex predators. The group included smaller bottom dwellers, filter feeders, and species adapted to specific niches. Some had flattened bodies suited to life near sediment surfaces. Others were more streamlined for active swimming in open water. This ecological diversity reflects adaptive radiation rather than a single dominant body plan. Internal skeletal structure in Placoderm shows transitional features between early jawless vertebrates and later bony fish. While external armour was prominent, internal skeletons consisted largely of cartilage reinforced with bone in certain regions. This hybrid composition reflects early vertebrate experimentation with skeletal materials. Sensory systems were well developed. Eye placement in many species suggests reliance on vision for hunting and navigation. The lateral line system, detecting water movement, likely complemented visual input. Combined sensory data improved prey detection and environmental awareness. The Devonian period is sometimes called the Age of Fishes because vertebrate diversity expanded significantly. Placoderms competed with early cartilaginous fish and early bony fish lineages. This competition encouraged further specialization in feeding and locomotion. Reef ecosystems during the Devonian supported high biodiversity. Placoderms exploited reef-associated prey populations, hunting among structural complexity. Reef collapse events later in the Devonian affected placoderm diversity in some regions. Energy requirements for large placoderms were substantial. Sustaining such predators required productive ecosystems with abundant prey. Devonian plankton productivity and invertebrate populations appear sufficient to support these demands. Isotopic analysis of placoderm fossils provides clues about trophic position. Carbon isotope ratios suggest many occupied upper levels of marine food webs. Injury and repair evidence in Placoderm fossils indicates intra-species competition as well as predator-prey interactions. Bite marks on armor plates demonstrate encounters with other large predators. Reproductive strategies among placoderms show intriguing diversity. Fossil evidence indicates internal fertilization in some species, with possible live birth in certain cases. This reproductive innovation enhances offspring survival compared with external spawning in predator-rich environments. Geographic distribution of placoderms was broad. Fossils appear on multiple continents, reflecting widespread marine habitats during the Devonian. Sea level fluctuations influenced habitat availability. Expanding shallow seas supported reef systems and coastal ecosystems where placoderms thrived. Oxygen levels during the Devonian were relatively high, supporting active vertebrate metabolism. Efficient circulatory systems allowed sustained swimming and rapid bursts during hunting. Climate variability toward the late Devonian introduced stress. Cooling events and anoxic conditions in certain marine basins contributed to declines in placoderm populations. Competition with emerging shark-like cartilaginous fish intensified as the Devonian progressed. Sharks possessed lighter skeletons and flexible bodies, potentially offering locomotor advantages. Placoderm armor provided excellent protection, but came with trade-offs. Heavy plating increases energy costs during swimming and reduces flexibility in some directions. Functional morphology studies reveal efficient jaw closure mechanisms. Lever systems within the skull maximize bite force relative to muscle size. Coprolites attributed to large Devonian fish sometimes contain fragments of bone and shell, supporting active predation. Predation pressure from placoderms influenced prey defensive strategies. Thickened shells and spines among invertebrates reflect continued arms race dynamics. Sedimentary deposits containing placoderm remains often show association with reef or nearshore environments, indicating preferred habitats. Evolutionary turnover within placoderms occurred throughout the Devonian. Early forms differ significantly from later large arthrodires. Mass extinction events near the end of the Devonian significantly reduced placoderm diversity. Environmental changes included widespread anoxia and climate shifts. Despite their extinction, placoderms represent an important evolutionary stage. They demonstrate that vertebrates could achieve dominance in marine ecosystems before the rise of modern fish lineages. Comparisons with modern armoured fish are limited because placoderms left no direct descendants. However, their anatomical innovations influenced later vertebrate evolution indirectly. The Devonian marine world thus hosted ecosystems where vertebrate predators rivaled or exceeded arthropod giants in size and impact. Food web complexity reached new levels. Multiple predator guilds coexisted, each with distinct hunting strategies. Biogeochemical cycling responded to increased vertebrate biomass and predation intensity. Habitat partitioning reduced direct competition. Some placoderms specialized in benthic zones, others in pelagic regions. Growth patterns in fossilized plates allow estimation of age and life history traits. Temperature gradients influenced regional distribution, with tropical zones supporting greater diversity. The decline of placoderms coincided with diversification of other vertebrate groups that would later dominate. Understanding placoderm ecology clarifies how vertebrate predation matured in Paleozoic oceans. Their presence illustrates that armour and aggression could coexist within a single lineage, combining defensive protection with offensive capability. As environmental instability increased toward the late Devonian, ecosystems faced significant restructuring, and examining how mass extinction events reshaped marine communities helps explain why even dominant armoured giants eventually disappeared from the fossil record. By the time the Devonian period was drawing toward its later stages, Paleozoic oceans had already experienced hundreds of millions of years of gradual escalation. Predators had become faster and more specialized. Defensive armor had thickened. Reef systems had expanded and matured. Vertebrate lineages such as placoderms had risen to dominance in many marine environments. Ecosystems were layered, competitive, and densely interconnected. It might seem, looking at that steady trajectory, that marine life was simply progressing in one continuous upward direction. But the geological record does not show uninterrupted growth. It shows periodic interruptions so severe that entire ecological structures collapsed. Mass extinctions are not minor fluctuations in species numbers. They represent intervals when a significant percentage of global biodiversity disappears within a relatively short geological span. In marine systems, these events can eliminate dominant predators, reef builders, plankton communities, and entire functional groups simultaneously. When that happens, oceans do not simply return to previous conditions, they reorganize. The late Ordovician extinction provides one of the earliest clear examples. Near the end of the Ordovician period, global climate shifted toward cooling. Evidence suggests glaciation occurred on parts of the Gondwanan supercontinent. When ice sheets expand, sea levels drop. Shallow epicontinental seas, which had supported immense biodiversity, contract dramatically. Habitat loss alone can eliminate numerous species. Reduced sea level also alters circulation patterns, salinity gradients, and nutrient distribution. Cooling affects ocean chemistry. Colder water holds more dissolved oxygen, but rapid climate change can disrupt established ecosystems before organisms adapt. Additionally, glaciation influences carbon cycling. Shifts in atmospheric carbon dioxide alter ocean acidity and temperature stability. Fossil records show significant marine biodiversity loss during this interval, particularly among shallow water organisms dependent on stable conditions. After the ice retreated and climate stabilized, surviving lineages diversified again. That pattern illustrates a core feature of mass extinctions. They clear ecological space. Vacant niches invite rapid evolutionary experimentation. However, the resulting ecosystems are not identical replicas of previous ones. Dominant groups often shift. The late Devonian extinction unfolded more gradually but proved equally transformative. Evidence indicates multiple pulses of extinction rather than a single catastrophic event. Widespread marine anoxia, meaning depletion of oxygen in deep waters, appears to have played a major role. Anoxic conditions are particularly destructive in marine environments because most complex animals depend on oxygen for metabolism. Anoxia can result from several interacting factors. Increased nutrient runoff from expanding terrestrial plant life may have stimulated plankton blooms. When large plankton populations die and decompose, oxygen is consumed in the process. Stratified water columns prevent oxygen replenishment from the surface. Deep water organisms suffocate. Reef systems collapse when key builders disappear. Placoderms, which had thrived during the Devonian, did not survive these environmental stresses. Their extinction did not occur because they were inherently flawed predators. Rather, ecosystem restructuring removed the conditions that had supported them. New vertebrate groups, including early sharks and bony fish, expanded afterward. The most severe of all Paleozoic marine extinction events occurred at the end of the Permian period, approximately 252 million years ago. Often called the Great Dying, this event eliminated an estimated 80 to 90% of marine species. The scale of loss far exceeded earlier crises. Reef ecosystems vanished almost entirely for millions of years. Trilobites, which had persisted for hundreds of millions of years, disappeared completely. Multiple factors likely contributed to the Permian extinction. Massive volcanic eruptions in what is now Siberia released enormous quantities of greenhouse gases, including carbon dioxide and methane. Rapid warming followed. Elevated temperatures reduce oxygen solubility in seawater. At the same time, increased atmospheric carbon dioxide dissolves into oceans, forming carbonic acid and lowering pH. Ocean acidification weakens organisms that rely on calcium carbonate shells and skeletons. Warming also intensifies stratification. When surface waters become warmer and less dense, they mix less with deeper waters. Reduced mixing limits oxygen delivery to depth. Anoxic conditions expand. Some geochemical evidence suggests hydrogen sulfide may have accumulated in certain basins, creating toxic conditions. Food webs collapse when foundational species disappear. Plankton declines affect filter feeders. Loss of filter feeders affects predators. The interconnected nature of marine ecosystems means cascading failure spreads rapidly once key nodes are removed. Extinction is selective to some degree. Organisms with broad environmental tolerance, high reproductive output, or flexible diets often fare better. Specialized reef builders or narrowly adapted predators may suffer disproportionately. Fossil evidence from post-extinction intervals shows simplified ecosystems dominated by opportunistic species. These so-called disaster taxa exploit vacant niches rapidly. Biodiversity remains low initially but recovers gradually over millions of years. Recovery patterns vary by region. Some basins stabilize sooner than others depending on local environmental conditions. Reef rebuilding after the Permian extinction required entirely new groups of organisms. Carbon isotope records reveal dramatic shifts during extinction intervals. Sharp negative excursions in carbon isotopes often indicate massive carbon release into the atmosphere-ocean system. Mass extinctions influence evolutionary direction as much as gradual selection does. By removing dominant lineages, they allow previously minor groups to expand. After the Permian extinction, new vertebrate and invertebrate groups would eventually dominate Mesozoic seas. It is important to recognize that extinction does not represent complete biological reset. Surviving lineages carry evolutionary history forward. Genetic diversity bottlenecks may reduce variation temporarily, but adaptive potential remains. Environmental thresholds matter. Gradual changes can accumulate without catastrophic effect until a tipping point is reached. Beyond that threshold, feedback loops accelerate collapse. Comparisons with modern ocean conditions sometimes draw attention to similar stressors: warming, acidification, oxygen depletion. Paleozoic extinction events demonstrate how sensitive marine systems can be to rapid chemical and temperature shifts. The concept of oceans resetting themselves does not imply intention or purpose. Reset refers to ecological restructuring after large-scale loss. Ecosystems reorganize around surviving lineages. Sedimentary layers often show abrupt shifts in fossil assemblages across extinction boundaries. Below the boundary, diverse reef communities. Above it, simplified assemblages dominated by fewer species. Mass extinctions also affect evolutionary tempo. Following large losses, speciation rates often increase as survivors diversify into available niches. Predator-prey dynamics change significantly after extinctions. Removal of apex predators allows certain prey populations to expand temporarily. Reef collapse during the Permian left a prolonged gap in carbonate platform development. It took millions of years for new reef builders to establish comparable structures. Marine invertebrate groups such as brachiopods declined after the Permian, while bivalve mollusks rose to prominence in later eras. Geochemical modelling helps reconstruct ocean temperature and oxygen distribution during extinction intervals. Tectonic activity and volcanic emissions interact with climate systems. The Siberian Traps eruptions illustrate how geological processes can trigger biological crises. The Paleozoic thus contains multiple examples of ecological rise followed by abrupt contraction. Understanding these extinction events clarifies that dominance, size, or defensive strength do not guarantee long-term survival. Placoderms, trilobites, reef builders, and countless other organisms once flourished, yet environmental change exceeded their adaptive limits. Each extinction event altered evolutionary pathways permanently. Marine ecosystems that emerged afterward differed structurally and compositionally from those before. The pattern of escalation, collapse, and reorganization recurs throughout Earth's history. Recognizing how oceans reset themselves helps explain why the Paleozoic, despite producing Earth's first marine giants and armored predators, ultimately gave way to entirely new marine communities that would define the next chapters of ocean evolution. When looking back at the Paleozoic oceans and the unusually large arthropods, armoured fish, and formidable predators that emerged there, it is tempting to focus only on anatomy and evolutionary competition. However, physical and chemical conditions set firm boundaries on what bodies can achieve. Oxygen availability, climate stability, and ocean chemistry collectively determine metabolic limits, structural possibilities, and ecological productivity. Without the right environmental framework, large-bodied organisms cannot persist regardless of evolutionary innovation. Oxygen plays a central role in determining body size. All complex animals rely on oxygen for cellular respiration, the process that converts nutrients into usable energy. Larger bodies require more energy to maintain tissues, power muscles, and sustain neural activity. In aquatic environments, oxygen enters the body through gills or diffuses across respiratory surfaces. The efficiency of this process depends on oxygen concentration in the surrounding water and the design of respiratory structures. During certain intervals of the Paleozoic, atmospheric oxygen levels were higher than at many other times in Earth's history. Higher atmospheric oxygen increases dissolved oxygen in surface waters, especially in well-mixed shallow seas. Elevated oxygen availability supports higher metabolic rates. Arthropods in particular are sensitive to oxygen concentration because many rely partly on diffusion-based respiratory systems. Higher oxygen allows diffusion to supply sufficient oxygen even to relatively large bodies. This helps explain why some Paleozoic arthropods reach sizes rarely seen in modern oceans. Vertebrates benefit from oxygen increases as well. Efficient circulatory systems transport oxygen from gills to tissues, but baseline environmental oxygen concentration still sets maximum performance. Sustained swimming and rapid bursts during predation require continuous oxygen supply. Devonian seas, characterized by relatively favorable oxygen levels in many shallow regions, supported active jawed fish and large placoderms. Climate stability interacts with oxygen availability. Temperature influences oxygen solubility in water. Cold water holds more dissolved oxygen than warm water. However, warmer water increases metabolic rates in ectothermic organisms, allowing faster growth and activity. The balance between temperature and oxygen concentration determines metabolic scope, meaning the range between resting and maximum metabolic rates. During intervals when temperature remained moderate and oxygen remained sufficiently high, metabolic scope expanded. Expanded scope enables active predation, larger muscle mass, and faster growth rates. Conversely, extreme warming reduces oxygen solubility while increasing metabolic demand, narrowing metabolic scope and stressing large organisms. Ocean circulation patterns strongly affect oxygen distribution. Well-mixed shallow seas replenish oxygen continuously. Stratified water columns limit mixing, allowing deep waters to become oxygen poor. Anoxic conditions in deeper basins restrict large animal habitation. Paleozoic epicontinental seas often provided well-oxygenated environments conducive to larger body sizes. Chemistry influences structural materials. Calcium carbonate saturation determines ease of shell and skeletal formation. During parts of the Paleozoic, seawater chemistry favoured calcite precipitation. This condition supports organisms constructing heavy armour or thick shells. If seawater chemistry shifts toward undersaturation, building and maintaining mineralized structures becomes energetically costly. Carbon dioxide concentration also affects ocean acidity. Elevated carbon dioxide lowers pH, increasing dissolution of calcium carbonate. Acidification weakens shells and skeletons, limiting defensive and structural potential. Stable pH conditions in certain Paleozoic intervals allowed extensive biomineralization, supporting armored organisms and reef builders. Nutrient availability underpins the entire system. Phosphorus, nitrogen, and trace minerals enter oceans through weathering and volcanic activity. Nutrient-rich waters support abundant plankton. High plankton productivity fuels food webs capable of sustaining large predators. Without a productive base, top predators cannot achieve significant size. Primary productivity determines energy transfer efficiency up the food chain. Energy diminishes at each trophic level. For a large predator to exist, lower trophic levels must generate sufficient biomass. Paleozoic shallow seas, with high productivity, created this energetic foundation. Climate influences continental weathering rates. Warmer, wetter climates accelerate chemical weathering of rocks, increasing nutrient runoff into oceans. Increased nutrients boost productivity, but can also trigger oxygen depletion if excessive. Seasonal variation affects growth patterns. Stable climates with predictable seasonal cycles allow consistent reproduction and feeding. Extreme fluctuations create stress, limiting maximum body size. Atmospheric composition interacts with ocean chemistry continuously. Carbon burial in marine sediments reduces atmospheric carbon dioxide, affecting global temperature. Shell-producing organisms contribute to long-term carbon cycling. Salinity stability supports metabolic efficiency. Large organisms often tolerate narrower salinity ranges. Fluctuating salinity stresses physiology and limits growth. Tectonic activity influences ocean basin geometry. Wide continental shelves promote shallow marine habitats. Narrow shelves restrict them. Habitat area influences total ecosystem biomass capacity. Energy cost of maintaining large armor or thick exoskeletons is significant. Environmental chemistry must support efficient mineral acquisition for such structures to remain viable. Evolutionary potential interacts with environmental opportunity. Genetic mutations enabling larger body size persist only if environmental resources support them. Predator-prey dynamics scale with size. Large predators require proportionally larger or more abundant prey. Ecosystem productivity must scale accordingly. Oxygen minimum zones, even if localized, restrict vertical distribution of large animals. Many Paleozoic giants likely remained in well-oxygenated upper waters. Thermal stratification can isolate surface ecosystems from deeper nutrient reservoirs. Upwelling currents mitigate this by bringing nutrients upward. Devonian reef systems illustrate interplay between chemistry and biology. Carbonate saturation supported massive reef construction, which in turn influenced local water flow and oxygen distribution. Mass extinction events often correlate with chemical disruptions. Ocean acidification, warming, and oxygen depletion during extinction intervals reduced maximum body size potential. Following extinction, surviving lineages often consist of smaller, generalist species better suited to unstable conditions. Comparisons with later geological periods show repeated correlations between oxygen peaks and increases in maximum body size among certain groups. Marine arthropod gigantism during high oxygen intervals reflects physiological constraints being relaxed. Bone mineralization in vertebrates also depends on calcium availability in stable pH conditions. Ecological carrying capacity depends on both resource supply and environmental stability. Stable intervals permit gradual body size increase through selection. Temperature gradients across latitudes influence regional size patterns. Some evidence suggests larger individuals in certain cooler, oxygen-rich waters. Reproductive strategy interacts with size. Larger animals often produce fewer offspring but invest more energy in each. Biogeochemical feedback loops connect organism size to environmental chemistry. Larger animals influence nutrient cycling through waste production and predation patterns. Sediment oxygenation from burrowing species supports more diverse benthic communities, indirectly supporting larger predators. Ultimately, size is constrained by physics and chemistry as much as by evolutionary competition. Mussels require oxygen, skeletons require minerals, food webs require productivity. Paleozoic oceans provided intervals where these constraints aligned favourably, allowing unusually large arthropods and armoured vertebrates to thrive. When oxygen dropped, climates shifted, or chemistry destabilised. Maximum size potential contracted accordingly. Understanding these environmental controls clarifies that Paleozoic monsters were not anomalies but predictable outcomes when ecological opportunity intersected with favourable planetary conditions. And examining how fossil evidence preserves these patterns helps connect environmental chemistry directly to the physical remains that reveal the scale of life in those ancient seas. When people picture prehistoric oceans, the image often comes from fossils displayed in museums or reconstructed in documentaries. Shells, armoured fish plates, trilobite exoskeletons, reef limestone, and other mineralised remains dominate that picture. It creates a strong impression that ancient marine life was mostly hard, spiny, and heavily armoured. The reality is more balanced. Soft-bodied organisms almost certainly made up a large proportion of Paleozoic ecosystems, but preserving soft tissue over hundreds of millions of years requires unusually specific conditions. Understanding how fossils form helps explain why certain animals appear frequently in the record, while others remain comparatively rare. Fossilization begins with burial. When an organism dies, decomposition normally starts quickly. Scavengers, bacteria, chemical breakdown, and physical disturbance all act to destroy tissues. For fossilization to occur, burial must happen before complete decay. Sediment carried by currents, storms, river input, or underwater landslides can rapidly cover remains. Fine-grained sediments such as mud or silt are particularly effective because they reduce oxygen exposure and slow decomposition. Hard tissues fossilize more easily because they resist decay. Shells made of calcium carbonate, bones composed of mineralized tissue, and arthropod exoskeletons reinforced with chitin and minerals persist longer in sediment. Even so, they may dissolve or fragment depending on environmental chemistry. Soft tissues, lacking mineral reinforcement, usually disappear unless preservation conditions are exceptional. Exceptional fossil sites exist where unusual circumstances preserved soft-bodied organisms in detail. One of the best-known examples is the Burgess Shale in Canada. There, fine mudslides rapidly buried marine communities, sealing organisms in low-oxygen conditions that slowed decay. Over time, compression preserved outlines of soft tissues, digestive systems, appendages, and even eye structures. Without such sites, knowledge of early marine ecosystems would be far less complete. Another important locality is the Chengjiang biota in China, which similarly preserves soft-bodied Cambrian organisms. These sites demonstrate that early marine ecosystems included far more diversity than hard fossils alone suggest. Worm-like organisms, soft arthropods, jellyfish-like creatures, and other delicate animals become visible in these rare deposits. Mineral replacement plays a key role in fossil preservation. During diagenesis, minerals dissolved in groundwater infiltrate buried tissues. Original biological material may be replaced gradually by silica, calcite, or other minerals. This process preserves external shape and sometimes microscopic structure. Carbonization is another pathway. Organic tissues compress under sediment pressure, leaving thin carbon films outlining body shapes. This method often preserves delicate features such as limbs or soft body contours. Permineralization differs slightly. Instead of replacing tissues, minerals fill internal spaces, reinforcing structures while retaining original material. Fossilized bone often forms this way. Mold and cast fossils provide indirect evidence. If an organism decays after burial, it may leave an impression in sediment. Later mineral infill creates a cast replicating external shape. Trace fossils expand understanding further. Footprints, burrows, feeding marks, and resting impressions reveal behaviour even without body fossils. Trilobite trackways, for instance, indicate locomotion patterns. Burrow complexity reveals sediment interaction. Coprolites, fossilised feces, offer dietary evidence. Shell fragments, bone pieces, or organic residues inside coprolites indicate predator-prey relationships. Chemical fossils, sometimes called biomarkers, consist of molecular remnants of ancient organisms. Certain organic compounds persist in sediments and indicate biological origin even when physical structures vanish. Isotope analysis provides environmental context. Ratios of carbon, oxygen, sulfur, and other isotopes reflect temperature, salinity, oxygen levels, and biological activity at the time of deposition. Sedimentary context matters greatly. Fine-grained anoxic environments preserve fossils differently from high-energy sandy environments. Reef settings preserve skeletal material abundantly but rarely soft tissue. Tectonic processes affect preservation. Heat and pressure during mountain building can destroy fossils. Metamorphism alters original mineral structures. Erosion exposes fossils at the surface. Many discoveries occur where natural weathering or human excavation reveals sedimentary layers. Dating techniques place fossils within geological time. Radiometric dating of volcanic ash layers above or below fossil beds provides absolute age estimates. Relative dating through stratigraphy establishes sequence. Paleontologists reconstruct entire organisms from partial remains using comparative anatomy. Knowledge of modern relatives helps interpret fragmentary fossils. Computer tomography scanning allows non-destructive examination of fossil interiors. CT scans reveal internal structures without physical preparation. Three-dimensional modelling reconstructs body shapes and movement capabilities. Biomechanical analysis estimates swimming speed, bite force, or locomotion efficiency. Taphonomy, the study of fossilization processes, helps interpret biases in the fossil record. Understanding which organisms fossilize easily prevents overestimating their ecological dominance. Soft-bodied plankton rarely fossilize directly, yet they formed the base of many food webs. Their presence is inferred from sediment chemistry and associated fauna. Preservation bias affects perception of size distribution. Large mineralized organisms appear disproportionately common in the record. Rare soft tissue preservation occasionally reveals pigmentation patterns, though interpretation remains cautious. Growth rings in shells or bones record seasonal environmental variation. These rings help reconstruct climate cycles. Fossil assemblages often represent communities rather than isolated individuals. Community composition informs ecosystem reconstruction. Mass mortality layers sometimes indicate sudden environmental events such as storms or anoxia. Experimental taphonomy studies how modern organisms decay under controlled conditions to improve fossil interpretation. Microfossils, including foraminifera and radiolarians, provide detailed environmental records due to their abundance and sensitivity to conditions. Paleoecology integrates fossil data with sedimentology and geochemistry to reconstruct ancient ecosystems comprehensively. Comparative stratigraphy across continents reveals global patterns in biodiversity and extinction events. Preservation of early fish cartilage is rare, but mineralization in certain conditions allows occasional discovery. Fossil reefs preserve community interactions, showing spatial relationships among organisms. Geochemical proxies help identify ocean acidity and temperature during fossil deposition. Fossil preparation techniques include mechanical removal of matrix, chemical dissolution, and laser imaging. Digital databases compile fossil occurrences globally, enabling large-scale biodiversity analysis. Understanding fossilization limitations prevents misinterpretation of evolutionary timelines. Soft-bodied organisms may have played ecological roles equal to or greater than those of heavily armoured species. Reconstructing ancient food webs requires integrating multiple fossil types. Paleozoic fossil evidence remains incomplete, but continually expanding with new discoveries. Advances in imaging technology improve detection of subtle features. Interdisciplinary approaches combining geology, chemistry, biology, and physics enhance accuracy. The fossil record is a filtered archive rather than a complete catalogue. Recognizing that filter helps explain why Paleozoic oceans appear dominated by armoured monsters, even though softer life forms were abundant. Every fossil discovery adds detail to the broader picture of ancient marine ecosystems. Understanding how preservation works connects environmental conditions directly to what evidence survives today. That connection becomes especially important when considering behaviour and interaction patterns because fossils rarely capture action directly. Yet careful interpretation of preserved structures, traces, and context allows reconstruction of how these ancient marine organisms actually lived, hunted, avoided predators, and interacted within their environments. When reconstructing life in Paleozoic oceans, anatomy alone does not tell the full story. Fossils preserve shells, bones, armour plates, and occasionally impressions of soft tissue, but behaviour itself rarely fossilizes directly. Yet behaviour is what determines how organisms interact, how predators hunt, how prey avoid capture, how food webs operate, and ultimately how ecosystems function day to day. Scientists piece together behavioural patterns by combining anatomical evidence, trace fossils, sediment context, modern biological comparisons, and biomechanical modelling. This layered approach allows surprisingly detailed insight into how prehistoric marine animals likely lived. Predation remains one of the clearest drivers of behavioural evolution. Once active hunters appeared, prey species began adopting avoidance strategies. Enrollment behaviour in trilobites offers a clear example. Fossils preserved in curled positions suggest this defensive posture was routine. The mechanics are straightforward. By curling into a ball, trilobites exposed only hardened exoskeleton surfaces, reducing vulnerability. Some species evolved spines that made the enrolled form even more difficult to manipulate. Burrowing behaviour also expanded during periods of increased predation. Trace fossils show increasingly complex burrow systems throughout the Paleozoic. Burrowing serves several purposes. It provides shelter from predators, stabilizes body temperature in fluctuating conditions, and offers access to organic rich sediment layers. Some burrows display branching patterns, suggesting extended habitation rather than temporary refuge. Ambush predation likely became common among larger arthropods and early jawed fish. Structural reef environments provided ideal concealment. Predators could remain motionless until prey approached within striking distance. Modern marine ecosystems show similar strategies among cephalopods, crustaceans, and certain fish species, supporting this interpretation. Active pursuit hunting also developed. Streamlined body shapes in placoderms and early sharks suggest sustained swimming capability. Efficient propulsion systems, including tail-driven locomotion and stabilizing fins, allow predators to chase prey rather than rely solely on surprise. Vision played a central role in both hunting and avoidance. Organisms with well-developed eyes could detect movement quickly. Prey species likely evolved rapid escape responses triggered by visual stimuli. Neural reflex pathways supporting such responses leave indirect evidence in anatomical structures. Chemical sensing complemented vision. Many marine organisms detect dissolved organic molecules released by injured prey or predators. While direct fossil evidence for chemical sensing is absent, anatomical features consistent with chemoreception appear in some fossil groups. Group behaviour probably emerged in multiple lineages. Fossil assemblages showing clusters of similarly sized individuals suggest schooling or aggregation. Group living reduces individual predation risk through dilution effect and increased vigilance. Migration patterns may also have existed. Seasonal changes in temperature, oxygen levels, or food availability could prompt movement between habitats. Sediment layers containing repeated species assemblages suggest cyclical environmental occupation. Feeding strategies diversified considerably. Deposit feeders processed sediment for organic material. Filter feeders extracted plankton from water columns. Grazers consumed microbial mats or algae. Predators targeted mobile prey. Each strategy required distinct behavioral adaptations. Reproductive behavior remains challenging to infer, yet certain fossil evidence offers clues. Egg clusters associated with specific sediment types suggest protected spawning grounds. Some Placoderm fossils indicate internal fertilization, implying more complex mating behavior than simple broadcast spawning. Juvenile survival strategies likely differed from adults. Smaller size increases vulnerability. Nursery habitats, shallow protected waters, or reef structures may have served as developmental refuges. Molting behavior in arthropods introduces predictable vulnerability periods. Fossil accumulations of shed exoskeletons suggest synchronized molting events, possibly reducing individual predation risk through sheer numbers. Camouflage probably evolved alongside visual predation. Sediment-colored exoskeletons, disruptive patterns, or structural mimicry would enhance concealment. While color rarely fossilizes, shell ornamentation patterns sometimes hint at camouflage functions. Territorial behavior may have developed in resource-rich reef environments. Competition for space among filter feeders and grazers suggests possible exclusion zones. Sound and vibration detection likely influence predator-prey interactions. Water transmits pressure waves efficiently. Organisms capable of detecting subtle vibrations gain early warning. Energy conservation strategies also shaped behavior. Continuous active hunting requires high energy intake. Some predators likely alternated between active and ambush strategies depending on prey availability. Vertical habitat partitioning reduced competition. Some species occupied near surface waters. Others remained near the seafloor. Depth-related light and oxygen gradients influenced these distributions. Environmental disturbances such as storms or temperature shifts influence short-term behavioral adjustments. Shelter-seeking behavior during storms likely improves survival. Parasite-host interactions probably existed, although fossil evidence is limited. Shell deformities sometimes indicate parasitic influence. Healing scars on shells and bones provide evidence of survival after predatory attacks. Such scars indicate not only predation, but also resilience and recovery capacity. Competition among predators influenced hunting strategies. Specialization reduces direct competition. Some predators targeted armored prey, others soft-bodied organisms. Food web modeling based on fossil assemblages helps reconstruct energy flow patterns. Predator-prey ratios offer insight into ecosystem stability. Symbiotic relationships also contributed to survival. Some organisms likely lived in close association with reef builders for protection or feeding advantages. Metabolic rates influence daily activity cycles. Warmer waters often correlate with increased activity levels among ectothermic organisms. Sediment oxygen levels influenced burrowing depth. Oxygen-poor layers limit habitation depth for many species. Behavioral plasticity increases survival under fluctuating conditions. Species capable of adjusting feeding or habitat use persist longer. Fossil trackways sometimes preserve directional movement patterns, suggesting purposeful navigation rather than random wandering. Coprolites reveal dietary diversity, indicating opportunistic feeding in some predators. Predator satiation strategies may have existed. Mass spawning events overwhelm predators temporarily, increasing offspring survival. Evolutionary arms races operate behaviourally as well as anatomically. Faster escape responses, improved sensory acuity, and altered feeding times all reflect behavioural adaptation. Climate-driven changes influence long-term behavioural evolution. Cooling events may have altered migration patterns. Habitat complexity generally correlates with behavioural diversity. Reef environments in particular encourage specialisation. Biogeographic isolation leads to regional behavioural variation, alongside anatomical divergence. The integration of anatomy, trace fossils, sedimentology, and modern analogues provides the most reliable behavioural reconstructions. No single fossil captures complete behaviour, but cumulative evidence produces coherent ecological pictures. Behavioural evolution often precedes structural change. New behaviours can create selection pressures favouring anatomical adaptation. Understanding prehistoric marine behaviour clarifies why certain lineages flourished while others declined. It also helps explain how ecosystems maintain stability despite intense predation. These behavioural dynamics set the stage for major evolutionary transitions, particularly when some marine vertebrates began exploring shallow coastal zones that occasionally exposed them to air, a shift that would eventually connect ocean ecosystems to terrestrial environments in ways that reshaped life on Earth permanently. For hundreds of millions of years, the oceans had been the primary stage for complex life. Arthropods had grown large beneath the surface. Jawed fish had diversified into predators and grazers. Reef systems had supported dense communities. Yet during the later Paleozoic, especially in the Devonian period, a gradual shift began that would permanently alter the distribution of life on Earth. Some vertebrates that had evolved entirely within marine environments started exploiting shallow freshwater habitats, and eventually the edges of land itself. This transition did not happen because the oceans failed. It happened because ecological opportunity expanded. Shallow coastal waters and estuaries formed dynamic environments. River systems carried sediments and nutrients from continents into the sea. Floodplains created seasonal ponds and lagoons. These transitional habitats fluctuated in salinity, oxygen levels, and water depth. Organisms capable of tolerating variability gained advantages in such unstable environments. Lobe-finned fish, particularly sarcopterygians, play a central role in this transition. Unlike ray-finned fish, whose fins consist primarily of thin bony rays, lobe-finned fish possess robust fleshy fins supported by internal bone structures. These bones share structural similarities with the limb bones of later terrestrial vertebrates. The presence of strong jointed fin bases allowed limited weight bearing in shallow water or on muddy substrates. Respiratory adaptation also contributed. Some early lobe-finned fish possessed primitive lung-like structures in addition to gills. These organs allowed supplemental air breathing. In oxygen-poor shallow waters, especially during seasonal drying, air-breathing ability provided survival advantage. Modern lungfish offer partial analogues for this dual respiratory strategy. Oxygen variability in Devonian freshwater systems may have been significant. Stagnant ponds can become hypoxic as organic material decomposes. Fish capable of gulping air avoid suffocation. Over time, reliance on air breathing could increase. Predation pressure in marine environments may also have influenced habitat exploration. Shallow, vegetation-rich freshwater habitats initially hosted fewer large marine predators. Reduced predation risk makes marginal environments more attractive despite physical challenges. Structural vegetation began spreading across terrestrial landscapes during the Devonian. Early vascular plants stabilized soils and created new ecological interfaces between land and water. Root systems altered sediment transport into rivers and coastal zones. Increased plant growth enhanced nutrient runoff, influencing freshwater productivity. The mechanical challenge of movement in shallow water differs from open water swimming. Weight bearing becomes more important as water depth decreases. Strong fin structures provide leverage against substrate. Over evolutionary time, fin bones elongated and joint articulation improved. Fossil species such as Tiktaalik illustrate transitional anatomy. Possessing both fish-like and tetrapod-like features, these organisms had robust fins capable of supporting body weight in shallow water. Their skull structure indicates flexible neck movement, useful in environments where head mobility aids feeding. Feeding strategies also adapted. In shallow or near-surface environments, prey types differ from open marine systems. Insects, small crustaceans, and other organisms inhabit water margins. Versatile jaw mechanics help exploit varied prey. Sensory adaptation accompanied structural change. Lateral line systems function best underwater. As organisms spent more time near or above water, visual and olfactory systems adjusted to new sensory conditions. Climate during parts of the Devonian supported extensive wetland development. Warm temperatures and high sea levels created broad coastal plains suitable for transitional habitats. Seasonal drought may have been an additional driver. Fish trapped in shrinking pools would benefit from short-distance terrestrial movement to reach other water bodies. Even limited locomotion across mud or vegetation provides survival advantage. The shift from water to land required multiple physiological adjustments beyond locomotion. Desiccation resistance becomes critical. Aquatic skin loses moisture rapidly in air. Gradual development of thicker skin or behavioral moisture retention strategies likely preceded full terrestrial adaptation. Gravity imposes new constraints. Buoyancy in water supports body mass. On land, skeletal strength must compensate fully. Strengthening of limb bones and vertebral columns follows logically. Reproductive strategy presents another barrier. Early tetrapod ancestors likely still relied on water for egg deposition. Fully terrestrial reproduction evolved later, with the development of amniotic eggs. Oxygen concentration in the atmosphere during late Paleozoic intervals was relatively high compared with earlier times. Higher atmospheric oxygen facilitates air breathing and supports terrestrial activity. Transition zones between fresh and marine water provided evolutionary stepping stones. Gradual salinity shifts allow incremental adaptation rather than abrupt environmental change. Competition in freshwater systems initially may have been lower than in established marine ecosystems. Reduced competition allows diversification. Predation continued shaping evolution. Early tetrapods faced new terrestrial predators eventually, but initial movement into marginal zones may have provided temporary refuge. Trace fossils, such as early tetrapod footprints preserved in Devonian sediments, provide evidence of weight-bearing locomotion on land. Comparative anatomy of fin and limb bones reveals homologous structures, confirming evolutionary continuity. Genetic evidence from modern vertebrates supports common ancestry linking fish and tetrapods. The transition was neither linear nor uniform. Multiple lineages experimented with shallow water lifestyles. Some returned fully to aquatic environments later. Marine ecosystems continued evolving independently. The emergence of land vertebrates did not halt ocean diversification. The movement onto land influenced marine systems indirectly. Increased plant growth altered carbon cycling and nutrient delivery. Devonian extinction events impacted both marine and freshwater lineages. Environmental instability shaped evolutionary outcomes on both fronts. Understanding this transition clarifies that Paleozoic oceans not only produced formidable marine predators, but also gave rise to the first vertebrates capable of colonizing land. The same environmental factors that supported marine gigantism, oxygen availability, nutrient productivity, structural complexity, also created opportunities in marginal habitats. From a broader ecological perspective, the shift illustrates how evolutionary innovation often begins at environmental boundaries. Oceans remained central to life, yet edges between ecosystems foster experimentation. Over millions of years, incremental changes in fin structure, respiration, and behaviour accumulated into fully terrestrial vertebrates. Those early land pioneers eventually diversified into amphibians, reptiles, mammals, and birds. The origin of land vertebrates thus traces directly back to marine ancestors shaped by Paleozoic ocean dynamics. Understanding that continuity emphasises that so-called ocean monsters were not evolutionary dead ends, but part of a branching tree that extended far beyond the sea. From this point, reflecting on how these ancient marine ecosystems continue influencing modern biodiversity and ecological structure helps connect Paleozoic history directly to the oceans that surround us today. Looking back at Paleozoic oceans can feel distant at first, almost disconnected from modern ecosystems. The creatures were unfamiliar, the continents arranged differently, the climates often warmer or more unstable. Yet when paleontologists, evolutionary biologists, and marine ecologists study these ancient organisms carefully, a consistent conclusion emerges. The same biological principles shaping life today were already operating then. Predator-prey feedback, environmental constraints, competition for resources, sensory adaptation, and reproductive strategy all worked in recognizable ways. The difference lies mainly in which groups happen to dominate at a given time. One of the clearest lessons concerns evolutionary arms races. The escalation between predators and prey visible in Paleozoic oceans continues in modern ecosystems. When early arthropod predators evolved grasping appendages, prey developed thicker shells and spines. Later, jawed fish evolved crushing jaws, and prey responded again with stronger armour or improved escape behaviour. Today, similar patterns exist. Crabs evolve stronger claws, mollusks evolve thicker shells. Fish improve swimming efficiency, prey fish refine schooling behaviour. The principle remains consistent. Adaptation rarely stops because interacting species continually influence each other. Environmental limits also remain fundamental. Paleozoic gigantism among arthropods and armoured fish correlates strongly with oxygen availability, climate stability, and ocean chemistry. Modern studies show similar relationships. Regions with high oxygen and nutrient productivity support larger marine animals. When oxygen levels decline, maximum body size often decreases. Climate-driven ocean deoxygenation observed today raises questions directly connected to those ancient patterns. Another important insight involves biodiversity resilience. Paleozoic oceans experienced multiple mass extinction events. Entire dominant groups disappeared, yet life persisted. Recovery took millions of years, but ecosystems eventually re-established complexity. This demonstrates both resilience and vulnerability. Marine ecosystems can recover from major disruptions, but recovery operates on geological timescales, far beyond human lifetimes. The fossil record also highlights contingency and evolution. Arthropods dominated early marine predator niches before vertebrates rose. Placoderms became major vertebrate predators before later fish groups replaced them. These transitions were not predetermined. Small environmental shifts, genetic innovations, or extinction events redirected evolutionary pathways. Modern biodiversity reflects similar contingency. Dominant groups today may not remain dominant indefinitely. Sensory evolution provides another continuity. The development of vision in Paleozoic seas reshaped ecological interaction. Modern marine ecosystems still revolve heavily around sensory capabilities. Visual predators dominate well-lit waters, chemosensory specialists thrive in murky or deep environments. Acoustic communication among marine mammals represents another sensory adaptation layered onto older systems. Habitat complexity continues driving biodiversity. Paleozoic reefs created dense ecological networks long before modern coral reefs appeared. Structural complexity fosters niche diversification, reduces direct competition, and stabilizes ecosystems. Contemporary reef conservation emphasizes the same principle. Habitat structure underpins biodiversity. Reproductive strategy diversity observed in ancient marine organisms also persists today. Broadcast spawning, egg brooding, live birth, and complex mating behaviors all appeared in early forms. Evolution rarely discards effective strategies entirely. Instead, it modifies them for new conditions. The transition from marine to terrestrial vertebrates provides a broader evolutionary lesson. Major ecological shifts often begin at environmental boundaries. Shallow coastal zones allowed incremental adaptation from water to land. Modern evolutionary research shows similar boundary-driven innovation in estuaries, mangroves, and other transitional habitats. Fossil evidence also underscores the importance of generalist species. During extinction events, organisms with flexible diets and broad environmental tolerance often survive better than highly specialized forms. Modern ecological studies confirm this trend. Generalists frequently adapt more readily to rapid environmental change. Energy flow principles remain unchanged. Paleozoic food webs depended on primary productivity from plankton and algae. Today's oceans still rely on microscopic photosynthetic organisms at the base of food chains. Changes in plankton populations cascade upward through ecosystems. Biogeochemical feedback loops observed in the Paleozoic continue shaping modern climate. Shell-building organisms sequester carbon. Plankton influence atmospheric carbon dioxide. Ocean chemistry and biology interact continuously. Another continuity involves ecological succession. After disturbances, ecosystems rebuild gradually. Early colonizers stabilize conditions. More specialized species follow. Fossil reef recovery after extinction events mirrors modern reef regeneration following storms or bleaching, though on vastly different timescales. Predator diversity contributes to ecosystem stability. Paleozoic oceans with multiple predator types maintained balanced food webs. Modern marine ecology similarly emphasizes the role of apex predators in regulating ecosystem structure. Evolutionary experimentation visible in early fossil assemblages reminds us that many anatomical innovations appear repeatedly across unrelated groups. Eyes, jaws, streamlined bodies, and mineralized skeletons evolved independently multiple times. This suggests strong selective pressures guiding similar solutions. Climate sensitivity remains a central theme. Paleozoic extinction events often coincide with rapid climate shifts, ocean acidification, or oxygen depletion. Current climate research examines comparable stresses affecting modern oceans. Evolution does not aim toward complexity automatically. Periods of simplification follow environmental stress. Fossil records show reduced biodiversity after extinctions before gradual recovery. Human influence now represents a new variable. Unlike natural Paleozoic changes unfolding over thousands or millions of years, modern environmental changes often occur within decades. The fossil record provides context for potential long-term outcomes, but not exact predictions. Comparative genomics reinforces fossil insights. Genetic pathways controlling development, respiration, and sensory systems show deep evolutionary conservation. Behavioral adaptation remains as important as anatomical change. Early marine organisms modified behavior in response to predators and environmental stress. Modern species exhibit similar plasticity. Evolutionary success does not guarantee permanence. Trilobites thrived for hundreds of millions of years, yet eventually vanished. Dominance depends on environmental compatibility, not inherent superiority. Fossil evidence also highlights interconnectedness. Changes in ocean chemistry influence biology, which in turn influences climate. Feedback loops operate continuously. Marine ecosystems have repeatedly reorganized around new dominant groups without halting overall biodiversity growth long term. Studying ancient oceans helps calibrate expectations. It demonstrates both the durability of life and the fragility of specific ecosystems. Modern conservation efforts often draw indirectly from paleontological insights about resilience, tipping points, and recovery patterns. The presence of transitional fossils connecting marine and terrestrial vertebrates underscores evolutionary continuity rather than abrupt appearance. Ocean ecosystems today still carry structural legacies established in the Paleozoic, including trophic layering and reef-based biodiversity hotspots. Understanding ancient evolutionary patterns helps interpret modern biodiversity trends without assuming uniqueness in current processes. Biological innovation continues, though often subtle. Microevolutionary changes accumulate as they did in the Paleozoic. Recognizing these continuities provides a grounded perspective on evolution as an ongoing process rather than a completed historical phase. The ancient marine organisms, often labelled as monsters, were simply participants in that process, responding to environmental conditions using available biological tools. Seeing them in that context reduces mystery while increasing appreciation for the scale of evolutionary time. This perspective naturally leads toward reflecting on how echoes of those ancient oceans remain present in modern marine ecosystems, shaping biodiversity, climate interactions, and the ongoing story of life beneath the surface. If you trace the arc of Paleozoic oceans from their early quiet complexity through the rise of predators, the spread of reefs, the escalation of armour, the emergence of jawed fish, and the repeated reset of mass extinctions, a clear pattern emerges. Life does not move in a straight line. It expands, restructures, collapses, and reorganises. The creatures that once dominated those ancient seas were not anomalies. They were responses to environmental opportunity, chemical balance, oxygen availability, and ecological interaction. When conditions aligned, large armoured arthropods and powerful vertebrate predators flourished. When conditions shifted beyond their tolerance, they disappeared. The modern ocean carries structural similarities to those Paleozoic systems. Food webs still begin with microscopic photosynthetic organisms. Nutrient cycling still depends on plankton productivity and sediment interaction. Apex predators still regulate intermediate consumers. Reef systems still concentrate biodiversity. Oxygen levels and temperature still constrain body size and distribution. The same ecological mechanics operate, even though the dominant species have changed. Consider how oxygen shapes marine life today. Regions with strong upwelling and high nutrient productivity often support dense fish populations and large predators. Conversely, expanding oxygen minimum zones reduce habitable depth ranges for many species. Paleozoic intervals of oxygen abundance corresponded with arthropod gigantism and active vertebrate predation. That parallel reminds us that physical chemistry remains foundational. Climate influences marine ecosystems continuously. Temperature affects metabolic rate, growth speed, and reproduction timing. In the Paleozoic, warming and cooling cycles restructured communities repeatedly. Today, warming oceans alter migration patterns, breeding cycles, and geographic distribution. The fossil record demonstrates that marine systems are sensitive to temperature thresholds. Ocean chemistry also continues to matter. Shell-forming organisms rely on calcium carbonate saturation, just as their Paleozoic ancestors did. Acidification reduces their structural integrity. Reef systems built by different groups across geological time show similar vulnerability when chemistry destabilizes. The rise and fall of dominant groups in ancient oceans underscores that evolutionary success is temporary in geological terms. Trilobites persisted for hundreds of millions of years before disappearing. Placoderms dominated Devonian waters, before extinction removed them. Modern dominant marine groups may persist for long spans, but history shows that permanence is not guaranteed. Recovery patterns after extinction events provide additional perspective. Following severe losses, ecosystems rebuild gradually, often with new dominant lineages. Biodiversity eventually returns, but community composition changes. That lesson illustrates resilience without implying immunity. Behavioural strategies visible in the fossil record continue shaping modern survival. Schooling, camouflage, burrowing, ambush hunting, and migratory patterns remain effective responses to predation and environmental variability. The underlying logic of survival remains consistent across eras. Habitat boundaries still foster innovation. Just as shallow Devonian waters allowed vertebrates to explore land, modern estuaries and coastal wetlands host species adapting to variable salinity and fluctuating conditions. Transitional zones continue acting as evolutionary laboratories. Energy flow principles remain unchanged. Large predators require abundant prey, and abundant prey depend on primary productivity. Disruptions at the base propagate upward. Paleozoic food webs demonstrate this cascading dynamic clearly. Mass extinction events from the past show that rapid environmental change can overwhelm even highly adapted organisms. While modern changes differ in origin, the biological consequences follow similar rules. Oxygen depletion, warming, and acidification each exert measurable stress. Geochemical feedback loops also persist. Marine organisms influence atmospheric carbon through shell production and burial. Biological processes shape climate over long timescales. Understanding ancient marine ecosystems enhances interpretation of modern ecological data. Fossil records provide baseline context for natural variability and long-term trends. The label monster applied to large Paleozoic organisms reflects human perception more than biological function. In their own ecosystems, they were balanced participants, constrained by resource availability and environmental limits. Modern oceans still contain animals that might appear monstrous from a distant perspective. Large sharks, deep-sea cephalopods, enormous filter-feeding whales. Each occupies a niche shaped by the same evolutionary pressures that once shaped eurypterids and placoderms. The absence of trilobites or placoderms today does not signal failure. It signals transition. Evolutionary turnover is part of ecological continuity. Sediment layers preserve echoes of those transitions. Limestone reefs record vanished builders. Black shale layers record anoxic crises. Fossil assemblages record ecological restructuring. Scientific tools continue refining understanding. Isotope analysis, imaging technology, and comparative genomics expand insight into ancient marine systems. Public fascination with prehistoric sea creatures often centers on size and appearance. Yet the deeper story involves environmental interaction, chemical constraint, and adaptive response. Marine ecosystems today still respond dynamically to environmental shifts. Species migrate, adapt, or decline depending on tolerance thresholds. Studying Paleozoic oceans reinforces that life adapts within limits set by physics and chemistry. Those limits have remained constant even as species changed. Every modern marine organism carries evolutionary heritage traceable through millions of years of ocean history. The boundary between ancient and modern becomes less distinct when viewed through evolutionary continuity. Our present oceans are not separate from those early seas. They are successive versions shaped by cumulative change. Predator-prey relationships today mirror patterns established during early sensory and anatomical escalation. Reef complexity remains a central driver of marine biodiversity, just as in the Ordovician and Devonian. Oxygen fluctuations continue shaping distribution patterns, echoing Paleozoic constraints. Climate-driven extinctions in the past illustrate the scale at which environmental change influences biodiversity. The fossil record does not provide predictions, but it does offer perspective. It shows that ecological restructuring can occur rapidly relative to evolutionary time. It shows that dominance shifts repeatedly. It shows that survival favours adaptability over size or armour alone. The ancient seas produced formidable organisms because conditions allowed it. When conditions changed, ecosystems reorganised. The modern world reflects accumulated outcomes of those reorganisations. Beneath the surface of today's oceans, food webs, sensory systems and structural habitats still operate under principles established hundreds of millions of years ago. Recognising that continuity connects distant geological history to present marine life. The Paleozoic oceans are gone in form but not in influence. Their patterns persist in the living systems surrounding us now. And understanding those patterns provides a grounded awareness of how deeply the past continues shaping the present beneath every wave.